There is a number sitting inside medical databases across Canada that nobody has fully explained. It is not a dramatic number. It does not announce itself with fanfare or press releases. It is simply there, filed quietly inside provincial health records and blood donor registries, waiting for someone to ask the right question. And the question is this: why does Canada have one of the highest concentrations of Rh-negative blood in the entire Western Hemisphere? Not just slightly higher than its neighbours, meaningfully, statistically, stubbornly higher, in a region of the world where that blood type was supposed to be nearly absent before the arrival of Europeans. The Americas were supposed to be Rh-negative territory in only the most vanishingly rare sense. Indigenous populations across North and South America carried it at frequencies close to zero, and yet here is Canada, sitting at frequencies that rival parts of Western Europe, and nobody can fully agree on why. If you are watching this to fall asleep, that is exactly why I make these videos. Maybe subscribe and let me know where you are right now and what time it is. There is something about a mystery that has no clean answer that pulls at the mind in a particular way. Not the mystery of a crime where the solution exists and just needs finding. Not a puzzle where all the pieces are present and only the arrangement is unclear. This is a different kind of mystery. This is the kind where some of the pieces are missing, where some of the pieces may have dissolved into the ocean floor thousands of years ago, where some of the pieces are locked inside the bones of people buried under the permafrost of northern Canada and may never be extracted cleanly enough to tell the full story. This is the kind of mystery that sits at the edge of what archaeology can prove, what genetics can trace, and what history dares to suggest. Canada stretches across the top of North America like a vast, cold, mostly unexplored archive. Its forests are older than the nations that claim them. Its coastlines have been walked by human feet for at least 15,000 years, and possibly much longer than that. Its rivers carry water that has been flowing since the last ice age ended, and the glaciers retreated north and the landscape slowly exhaled into something livable. And somewhere inside that deep human history, inside those thousands of years of migration and contact and survival and loss, the answer to the blood type question is hiding. The conventional story of North American prehistory goes something like this. Humans crossed from Siberia into Alaska via the Bering Land Bridge sometime between 15,000 and 20,000 years ago. They were fully Rh positive, because virtually every population that developed in East Asia carried the Rh protein. They spread south and east across the continent, adapting to every environment they encountered, and within a few thousand years had populated both North and South America, from the Arctic to the southern tip of Patagonia. Then Europeans arrived starting in the late 1500s, bringing with them the genetic diversity of a continent where Rh-negative blood had already been present for tens of thousands of years. The Rh-negative frequencies you see in Canada today are therefore a product of European colonization and nothing more. That is the conventional story. It is tidy, it is defensible. It is also, when you look closely at the actual data, deeply insufficient. Because the distribution of Rh-negative blood across Canada does not follow the pattern you would expect if European immigration alone explained it. It does not concentrate in the places where European settlement was densest and earliest, and then fade outward from those centres. The pattern is messier than that, stranger than that, and in certain specific regions of Canada so anomalously concentrated that the standard explanation begins to feel like someone trying to cover a complicated painting with a single coat of thin white paint. The shape of something older shows through. To understand why this matters, you have to understand a little bit about what the Rh factor actually is and why its distribution across human populations tells such an unusual story in the first place. Your blood is not a simple red liquid. It is an extraordinarily complex suspension of different cell types, proteins, and molecular markers that your immune system uses to distinguish self from non-self. Among those markers, the ones that matter most for transfusions and pregnancies are the ones that decorate the surface of your red blood cells. The ABO system, which gives you your A, B, AB, or O blood type, is one set of those markers. The Rh system is another. And within the Rh system, the marker that matters most is a protein called the Rhd antigen. If you have it on your red blood cells, you are Rh positive. If you do not have it, you are Rh negative. Most people on Earth are Rh positive. The global average sits somewhere around 85%. That means roughly 15% of humanity lacks the Rhd antigen entirely, and that 15% is not distributed evenly across the planet. It clusters in very specific places. The Basque region of Spain and France, where frequencies reach 30 to 35%. Western Europe generally, where it hovers between 15 and 25%, depending on the specific population. And then it drops dramatically as you move east and south. In sub-Saharan Africa, it is nearly absent. In China, Japan, and most of East Asia, frequencies fall below 1%. Among indigenous populations of Australia and the Pacific Islands, it is essentially non-existent. And among the pre-contact indigenous populations of the Americas, it was, by every measure we have, extraordinarily rare to the point of being virtually absent. Which is what makes Canada so strange. Because Canada has Rh-negative frequencies in the range of 15 to 17% overall, with certain regional populations pushing higher than that. Some Atlantic Canadian communities, some Quebec populations with deep Francophone roots, and some specific First Nations groups whose genetic histories are only beginning to be understood, all show numbers that do not fit neatly into the template of recent European immigration being the whole story. And here is where the mystery deepens into something genuinely unsettling. The people who should know the answer, the geneticists, the anthropologists, the historians, do not have a consensus. They have theories. They have competing frameworks. They have archaeological evidence that points in different directions. And they have, in some cases, a studied reluctance to go too far down certain lines of inquiry because the implications are enormous. If the Rh-negative frequencies in some Canadian populations significantly predate the period of documented European colonization, then something happened on this continent that we have not properly accounted for. Some form of contact, some form of migration, some ancient movement of people carrying that genetic marker arrived in North America well before Columbus and his successors and left a trace in the blood of populations that have been here ever since. That is not a fringe theory. That is a question being asked, carefully and with proper scientific rigor, by researchers working in genetics labs at universities across Canada and the United States. The fringe part is not the question. The fringe part is only the specific answers proposed by people who would rather slot the mystery into existing narratives, whether those narratives are about ancient astronauts or lost continents or secret bloodlines of European royalty transplanted to the New World. Strip away those colourful overlays and the core question remains solid. Why does Canada have so much Rh-negative blood, and where did it come from? To start trying to answer that, you have to go back a very long way. Further back than the Vikings who reached the shores of Newfoundland around 1,000 years ago. Further back than the Basque fishermen who were working the waters off the Grand Banks of Newfoundland by at least the early 1500s and possibly much earlier than that. Further back than any European whose name appears in a history book or whose voyage was recorded in a royal archive. You have to go back to a time when the shape of the North American coastline was completely different from what it is today, when the sea level was lower by hundreds of feet, when entire ecosystems that no longer exist stretched along shores that are now underwater, and when the people who lived along those shores left almost nothing behind except fragments of bone and the faintest whisper of their DNA in the bodies of their descendants. The end of the last ice age was not a gentle transition. Over a period of several thousand years, beginning roughly 14,000 years ago, the massive glaciers that had covered most of Canada began to melt. The process was not steady or gradual. It lurched. It paused. It lurched again. There were periods of rapid warming followed by centuries of relative stability, followed by sudden catastrophic floods as ice dams broke and released enormous volumes of fresh water into the oceans almost overnight. The sea level rose more than 400 feet over this period. Coastlines that had been inhabited for thousands of years, that had supported fishing communities and shellfish harvesters and maritime traders moving up and down the shore, were swallowed by the rising ocean. Villages that might have held the answers to our questions about pre-contact contact are sitting under several hundred feet of cold Atlantic water right now, and will likely stay there. This is not a small archaeological problem. This is a catastrophic loss of evidence. The coastlines of Atlantic Canada, particularly the area around Nova Scotia and the Bay of Fundy, and the shores of Newfoundland and Labrador, were among the most resource-rich environments on the continent for Ice Age human populations. The shallow, cold waters produced enormous quantities of marine protein. The kelp forests supported complex ecosystems. The coastal geography provided natural harbours and shelter from storms. These were places where people would have wanted to live, and where they almost certainly did live, for thousands of years before the seas rose and erased them. What this means is that the genetic signature of those early coastal populations, whatever it was, Whatever mixture of ancient migration routes and long-distance contacts and local adaptation had produced it is now largely invisible to archaeology. We cannot dig up those villages. We cannot analyze the bones of those ancient Canadians. We can only look at the living descendants of whoever survived the drowning of the coast and try to read backward through their genetics to understand where they came from and what their ancestors carried. And when geneticists have done exactly that, looking at the genetic profiles of indigenous populations from Atlantic Canada and the broader northeastern region, they have found things that do not fit the standard model of a single wave of migration from Siberia, populating the entire continent. They have found haplogroups, which are essentially genetic family trees defined by specific shared mutations that appear in North American populations but have very limited distribution in the East Asian and Siberian populations that supposedly gave rise to all Native Americans. They have found genetic markers with closer analogies to certain European populations than to Siberian ones. And in the blood type data, they have found those stubborn concentrations of Rh negative that refuse to be fully explained by the few centuries of European colonization that produced the modern multicultural country we call Canada. Let's spend a moment with the Basque because they are central to this story in ways that go far deeper than most Canadian history textbooks acknowledge. The Basque people are one of the oldest continuously inhabiting populations in Western Europe. Their language, Euskara, is a linguistic isolate, meaning it has no known relatives anywhere on Earth. It is not related to Spanish. It is not related to French. It is not related to any of the Indo-European languages that swept across Europe roughly 5,000 years ago, when agricultural populations from the steppes of Central Asia expanded westward and replaced or absorbed most of the indigenous languages of the continent. Euskara survived because the Basque survived, tucked into their mountain valleys between what is now northern Spain and southwestern France, genetically and culturally distinct in ways that have fascinated researchers for more than a century. And the Basque carry Rh negative blood at a frequency that is unmatched anywhere else on the planet. Depending on the specific community being studied, somewhere between 30 and 35% of Basque people are Rh negative. This is roughly twice the European average, and more than 30 times higher than the frequency found in most indigenous populations of the Americas. Whatever happened in the deep genetic past to produce this remarkable concentration of Rh negative blood happened in or around the Basque homeland, or it happened to the ancestral population that became the Basque, possibly during some ancient period when a small founding group carrying the Rh negative variant at high frequencies became isolated, and that frequency became locked in through genetic drift. But here is what most people do not know about the Basque, and what is directly relevant to the Canadian mystery. The Basque were not just mountain farmers. They were among the most skilled maritime people in the world during the medieval period and into the early modern era. Their shipbuilding traditions were sophisticated and ancient. Their knowledge of Atlantic waters was deep and intimate, and there is substantial evidence, both documentary and archaeological, that Basque fishermen were regularly working the waters off the coast of Atlantic Canada, well before the official date of European contact that most textbooks use. The Grand Banks of Newfoundland are one of the richest fishing grounds in the world. The shallow, cold waters that sit on the continental shelf off the coast of Newfoundland and Nova Scotia support enormous populations of cod, haddock, halibut, and a dozen other species. Basque fishermen had developed industrial-scale cod fishing operations in the Bay of Biscay, and by the time Columbus sailed in 1492, they were already probing the edges of the western Atlantic, following the fish. There is documentary evidence of Basque ships working the Newfoundland fishery by the early 1500s, but historians who have studied the trade patterns and ship movements carefully have argued convincingly that the Basque knew about these waters for decades before any document formally recorded their presence there. A site called Red Bay on the southern coast of Labrador has become one of the most important archaeological discoveries in Canadian history for this reason. Red Bay was a Basque whaling station, and when archaeologists excavated it starting in the 1980s, they found evidence of operations that must have been running for many decades before the site was eventually abandoned. There were whale oil processing facilities, ship repair equipment, and most importantly, burial sites. The bodies of Basque sailors who died on this Canadian shore were interred at Red Bay, and their remains have been studied in detail. Among the things that study revealed was a genetic profile consistent with Basque origin, including the Rh negative frequencies that define that population. These were not isolated visitors. These were people living and dying and being buried in Canada, running operations that employed hundreds of workers, maintaining infrastructure that required significant local knowledge and likely involved ongoing, sustained interaction with indigenous communities. The question is not whether the Basque came to Canada before Columbus. They did. The question is how long before and what the nature of their contact with indigenous populations was. And when you ask that question, you open a door into a series of connected mysteries that cascade outward in every direction. Consider the Mi'kmaq. The Mi'kmaq are an indigenous people who have inhabited the Atlantic coastal regions of Canada, particularly Nova Scotia, New Brunswick, Prince Edward Island, and parts of Newfoundland, for at least 10,000 years. They are one of the most extensively studied indigenous groups in northeastern North America, and their oral traditions, their material culture, and their genetic profiles have all been the subject of significant research. And the Mi'kmaq present a number of puzzles that are not easily resolved by the standard model of North American prehistory. Their language, Mi'kmaq, belongs to the Algonquian language family, which is one of the largest and most widely distributed language families in North America. But within that family, Mi'kmaq shows some peculiar features. Certain researchers have pointed to structural elements in Mi'kmaq that suggest influences or developments that are not present in related languages to the west and south, possibly indicating a period of relative isolation and independent development, or possibly indicating contact with populations that spoke entirely different languages and left a trace in the vocabulary and grammar. More concretely, Mi'kmaq oral traditions preserve accounts of contact with people who arrived from the sea, who were not indigenous to the continent, and who interacted with Mi'kmaq communities in ways that range from trade to conflict to intermarriage, well before the European contact events that are historically documented. These oral traditions have generally been treated by Western historians as legend or allegory, which is what Western historians tend to do with oral traditions when they conflict with the written record. But the oral traditions of indigenous peoples are increasingly being recognized by researchers as preserved historical memory, sometimes accurate to a degree that is surprising when tested against independent archaeological or genetic evidence. And the genetic evidence in the case of the Mi'kmaq is interesting. Studies of Mi'kmaq DNA have found haplogroups and genetic markers that are unusual by the standards of what would be expected from a purely Siberian origin indigenous North American population. Some of these unusual markers appear to be very old, not the result of recent European admixture, and not easily explained by the known migration routes from northeastern Asia. Where did they come from? The honest answer is that no one fully knows. The hypothesis of pre-Columbian Atlantic contact involving Basque or other European maritime populations remains unproven, but has not been definitively ruled out by the genetic data. And the Rh negative frequencies in communities with significant Mi'kmaq ancestry are, in some cases, higher than what you would expect from European colonial admixture alone. Now let's add another layer to this. The Norse. In the year 1000 CE, give or take a few decades, a Norse explorer named Leif Erikson sailed from Greenland to a place he called Vinland, which most historians now believe was located somewhere on the northern tip of Newfoundland. The archaeological site at Lansor Meadows in Newfoundland has been excavated and confirmed as a genuine Norse settlement, with radiocarbon dates placing its occupation squarely around 1,000 years ago. This is no longer controversial. The Norse were in Canada. They built structures there. They repaired boats there. They almost certainly interacted with indigenous peoples there, whom they called Skraelings in their sagas. What is still being debated is how extensive the Norse presence was, how long it lasted, and what genetic trace, if any, it left in indigenous Canadian populations. The Lansor Meadows site appears to have been a short-lived outpost, used perhaps seasonally for a period of decades before being abandoned. But the Norse sagas describe multiple voyages to Vinland, contacts with indigenous people that included both trade and violent confrontation, and at least one account of a Norse woman, Gudrid, spending significant time in the New World and possibly giving birth to a child there. Whether any of those Norse visitors remained long enough to intermarry with local populations and contribute to the gene pool is unknown, but the biological mechanism for such a contribution certainly existed. The Norse of that period were not carrying the same genetic profile as the Basque, but they were carrying a Northern European profile that included Rh negative frequencies in the 15 to 20% range. If any Norse individuals remained in North America and had children with indigenous women, as maritime contact history elsewhere in the world has shown to be a nearly universal pattern, that would have introduced Rh negative alleles into populations that previously carried none or almost none. But here is the complication. One or two Norse visitors having children with indigenous people would not explain the Rh negative frequencies we see in Atlantic Canada today. The mathematics do not work. The Norse settlement at Lanceau Meadows was not large enough or long-lasting enough to account for a meaningful shift in the genetic frequency of a protein marker across an entire regional population. If the Norse contribution was real, it was a small signal added to a larger one. Something else, something bigger, would need to explain the rest. And that something else might have deeper roots than any European contact, no matter how early. It might be embedded in the original peopling of the Americas itself. The Beringia land bridge story is taught in schools as a settled matter. People came from Siberia, crossed the land bridge, moved south and east, and populated the Americas. This is broadly true in the sense that it captures the dominant genetic lineage of Native American populations. The majority of Native American ancestry traces back to northeastern Asian populations, and the timing of the crossing appears to have been roughly 15,000 to 20,000 years ago. But the word majority is doing a lot of work in that sentence, and researchers working at the edges of this field have been accumulating evidence for decades that the story is considerably more complicated. There is a genetic signal in some Native American populations, particularly in South America and in specific northeastern North American groups, that does not appear to derive from the standard Northeast Asian source population that produced most Native American ancestry. It is an ancient signal, meaning it is not the result of post-Columbian European admixture. It appears to be genuinely old, and it seems to have some relationship to certain West Eurasian populations, which is a technical way of saying populations from Western Europe and the Middle East and adjacent regions, rather than East Asia. This genetic anomaly has been named the Anzic signal in some contexts, and the Clovis ancestry signal in others, after the Clovis culture that appears to have been widespread in North America around 13,000 years ago. The Anzic child, an infant buried in Montana roughly 12,600 years ago, and among the oldest securely dated human remains in the Americas, carried this unusual genetic component. And when researchers compared the Anzic child's genome to populations around the world, they found something startling. Part of the ancestry matched East Asian populations, as expected, but another part matched ancient West Eurasian populations in a way that was not explained by the post-Columbian European gene flow that has affected virtually every indigenous American population since the 1500s. How did West Eurasian ancestry get into a 12,000-year-old child buried in Montana? The honest answer is that scientists are still arguing about this. One theory is that there was a population somewhere in northeastern Siberia during the Ice Age that had been shaped by gene flow from both East Asian and West Eurasian sources, essentially a mixed population that then crossed into the Americas and contributed to the founding ancestry of Native Americans. This model does not require anyone to have crossed the Atlantic. It requires only that the Ice Age Siberian population was itself genetically diverse in ways that included West Eurasian ancestry from very ancient contact events that happened long before the Americas crossing. But another possibility, one that some researchers take seriously and others dismiss, is that the West Eurasian signal in ancient Native American populations reflects actual contact across the North Atlantic, either via a coastal route hugging the ice edge or via some more direct crossing at a time so ancient that it predates any written record or any historical memory by tens of thousands of years. This is sometimes called the Solutrean hypothesis, after the Solutrean culture that occupied parts of southwestern Europe, including the Basque region, roughly 20,000 to 15,000 years ago, during the peak of the last ice age. The Solutrean hypothesis proposes that some Solutrean people crossed the Atlantic, following the edge of the sea ice that connected Europe to North America during the glacial maximum, and contributed to the peopling of the eastern seaboard of North America. The Solutrean hypothesis is controversial. It is deeply controversial, actually, with most mainstream archaeologists and geneticists rejecting it as insufficiently supported by evidence. But it has not been definitively killed by the data either, and certain features of some very early North American archaeological assemblages, particularly a stone tool tradition found on the Chesapeake Bay coast and radiocarbon dated to around 19,000 years ago, bear a stronger resemblance to Solutrean European tools than to any known Asian tradition. You can see why this matters for the Rh-negative question. The Solutrean people, as a European population living in the same region where Basque populations later developed, would have carried Rh-negative blood at frequencies far higher than any northeastern Asian population. If any of them crossed the Atlantic, If any of their descendants survived and mixed with the populations that eventually became the indigenous peoples of eastern North America, they would have seeded a genetic legacy that included elevated Rh-negative frequencies. That legacy would have been very ancient. It would have been present in North America long before the Norse arrived, long before the Basque fishermen anchored in the waters of the Grand Banks. And it would explain why certain northeastern North American populations showed genetic anomalies that are difficult to account for by any later European contact. This is where the story pivots into something that most people, even Canadians with a deep interest in their country's history, have never heard about. Because underneath the more dramatic theories about Ice Age crossings and pre-Columbian Atlantic contact, there is a quieter and in some ways more compelling body of evidence that has been building for decades in the form of objects, not theories. Physical artifacts that show up in places where they should not exist, made of materials that should not have travelled there, at dates that should not have been possible given the conventional timeline of North American history. The most striking of these involves copper. Specifically, what archaeologists call the Old Copper Complex, which is a cultural tradition centered around the Great Lakes region of North America that produced copper tools, weapons, and ornaments of extraordinary quality and quantity beginning roughly 8,000 years ago and continuing for thousands of years. The people of the Old Copper Complex were not farmers. They were hunter-gatherers, living in a period long before any settled civilization had emerged in North America. And yet they were mining, processing, and working pure copper on an industrial scale that has no parallel anywhere else in the world at that time. The source of the copper was Lake Superior. The shores of Lake Superior, and particularly the Keweenaw Peninsula of what is now Michigan, contain some of the world's largest deposits of native copper, meaning copper that occurs in its pure metallic form rather than as ore that needs smelting. You can pick it up off the ground. You can hammer it into shape without any metallurgical knowledge at all. And the ancient peoples of the Great Lakes region discovered this resource and exploited it with a thoroughness that staggers the modern imagination. Mining pits have been found across the Keweenaw that show systematic extraction operations extending over centuries. Estimates of the total amount of copper removed by ancient people from this region range into the hundreds of millions of pounds. That is not a typo. Hundreds of millions of pounds of pure copper, extracted over thousands of years by people using stone hammers and wooden tools. And then it largely vanished from the archaeological record. The copper was taken out of the ground. The people who took it out used some of it locally. But archaeologists can account for only a fraction of the copper in the archaeological sites of the Great Lakes region. The rest went somewhere else. It was traded. It moved. It travelled in quantities that imply organised long-distance trade networks operating across North America and possibly beyond, at dates that predate any civilization on the continent by thousands of years. This matters for the Rh-negative question because of where the copper ended up. Copper objects from the Great Lakes have been found in archaeological sites from Ontario to Florida, from the Atlantic coast to the Western Plains. Trade networks that distributed this copper across the continent would have moved people along with goods, because ancient trade was almost never conducted by anonymous strangers passing commodities along a chain. Ancient trade was conducted by people who travelled, who spent time in distant communities, who formed alliances and sometimes marriages with the people they traded with. If the people running the northern end of these trade networks, the people mining and distributing the copper of Lake Superior, carried unusual genetic profiles, including elevated Rh-negative frequencies, those genetic profiles would have moved with the copper. Is there any evidence that the Old Copper Complex people had unusual genetic profiles? The honest answer is that we have very limited ancient DNA from this region and this time period. The acidic soils of the Great Lakes region are not kind to ancient biological material. What little ancient DNA has been extracted from remains associated with this cultural tradition has not yet been sequenced in enough detail to give us clear blood type information. But the physical characteristics of some of the Old Copper Complex burials, which are elaborate and suggest a society with significant social stratification, include skeletal features that some physical anthropologists have noted as atypical for purely Northeast Asian-derived populations. And here is an additional thread that connects back to the main narrative. The geographic distribution of Rh-negative blood in modern Canada does not simply follow the distribution of European origin populations in the way you would expect if European colonial immigration were the whole story. It is elevated across much of the country, yes, and the European immigrant population accounts for a large part of that. But when researchers have looked specifically at communities with documented indigenous ancestry and controlled for the degree of European admixture, the Rh negative frequencies in some of those communities remain higher than the East Asian baseline would predict. Not dramatically higher, in most cases, not anomalously, obviously higher in a way that would constitute proof of ancient pre-Columbian contact by itself. But persistently, stubbornly, slightly higher than the model would predict, in a way that accumulates across multiple independent datasets into something that is hard to wave away as noise. Let us now travel to a specific place and a specific community that represents one of the most concentrated versions of this anomaly. The Ile d'Orléans sits in the middle of the St. Lawrence River, just downstream from Quebec City. It has been inhabited continuously since the time of the first French settlement of Canada in the early 1600s. The families who settled there in the 1600s are in many cases the same families whose descendants still live there today. The population of the Ile d'Orléans has been one of the most intensively studied in the world from a genetic genealogy perspective, precisely because of this continuity and because the detailed parish records that were kept by the Catholic Church from the very earliest years of settlement allow researchers to trace family lineages back with an accuracy that is extremely rare in historical population genetics, and the Ile d'Orléans population has elevated Rh negative frequencies. The explanation that most researchers have offered is straightforward. The founders of this population were disproportionately drawn from specific regions of France, including regions in southwestern France that had historical Basque connections and higher than average Rh negative frequencies. The founding population effect, combined with the genetic drift that occurs in any small, relatively isolated community over many generations, amplified those frequencies to levels that are noticeable when compared to the broader North American baseline. This is probably true. It is almost certainly a significant part of the story. But even within this well-documented and thoroughly studied population, there are threads that don't quite tie off. The earliest French settlers who came to Quebec did not arrive in a completely empty landscape. They arrived in a landscape that had already been inhabited for thousands of years, that already had established populations with their own complex social structures and territorial claims, and that had already experienced at least limited contact with European maritime activity in the preceding century, including those Basque whalers and fishermen whose presence at Red Bay and elsewhere along the Atlantic coast had been ongoing for decades before any permanent French settlement was established. The indigenous people of the St. Lawrence Valley at the time of first contact included the St. Lawrence Iroquoians, a group who were present in large numbers when Jacques Cartier arrived in 1534 and 1535, but who had mysteriously disappeared by the time Samuel de Champlain established the first permanent French settlement at Quebec in 1608. Where they went is one of the most debated questions in Canadian history. The St. Lawrence Iroquoians were a sophisticated, sedentary agricultural people who maintained large towns along the river and had what appear to have been extensive trade networks reaching both east and west. And then they were simply gone. No mass graves explaining a sudden epidemic. No clear archaeological evidence of a military defeat so total as to eliminate an entire people, just absence where there had been presence. The dominant theory is that the St. Lawrence Iroquoians were dispersed, absorbed by warfare and disease and displacement into neighbouring groups over the 70 years between Cartier's visit and Champlain's arrival. This is plausible and probably substantially correct, but it means that whatever genetic legacy the St. Lawrence Iroquoians carried, whatever unusual markers might have been present in their blood from their own deep history, was dispersed into the broader indigenous population of northeastern North America and potentially, through the intermarriage that occurred between the earliest French settlers and indigenous women, into the founding European origin population of Quebec as well. The genetic history of early Quebec is complex in ways that are still being unravelled. The church records track legal marriages, but they do not always track the full complexity of biological parentage in a frontier society where men vastly outnumbered women in the early decades of settlement, where the fur trade brought French men into sustained and intimate contact with indigenous communities across an enormous geographic range, and where the offspring of those relationships occupied an ambiguous social position that the historical record often underrepresented or misrepresented entirely. The Métis, the people of mixed European and indigenous ancestry who developed a distinct cultural identity in the Canadian West, are the most visible legacy of this history, but they are not the only one. Across Quebec and Atlantic Canada, In communities that identify as French Canadian or Acadian, there are people carrying genetic profiles that reflect both European and Indigenous ancestry, sometimes in proportions that are not obvious from their family histories or cultural identities. And in those mixed genetic profiles, the Rh negative frequencies are sometimes higher than they should be if the European component were the only source of that marker. But we have been circling the Atlantic and the eastern part of the country for long enough. Canada is a vast place, and the Rh negative anomaly is not confined to the east. It shows up in patterns across the country that are interesting, if not always easy to interpret. And to understand those patterns, you have to understand something about the deep genetic history of Indigenous North America that goes well beyond the simple Beringia crossing story. The haplogroups, those genetic family trees defined by specific shared mutations in mitochondrial DNA or the Y chromosome, tell a story about North American prehistory that is considerably more complex than the standard model suggests. The major haplogroups found in Native American populations are labeled A, B, C, D, and X. The first four of those, A through D, have clear Northeast Asian origins. They are found in Siberian and East Asian populations and almost certainly came to the Americas with the founding populations that crossed Beringia. Their presence in Native American populations is exactly what you would expect from the conventional model. Haplogroup X is different. Haplogroup X is found in Native American populations, particularly in the Great Lakes region and northeastern North America. It is also found in Europe and Western Asia, but it is not found in Siberia or East Asia at any significant frequency. This is a puzzle. If all Native American ancestry came through Siberia, then all Native American haplogroups should have Siberian equivalents. Haplogroups A through D check that box. Haplogroup X does not. The presence of haplogroup X in northeastern Native American populations has been one of the most debated findings in the genetics of Native American prehistory. It was identified in the late 1990s and has been a source of controversy ever since. The conventional explanation is that haplogroup X did exist in ancient Siberian populations, but has since disappeared from that region through genetic drift and population replacement, while surviving in both the Americas and in Europe through separate lines of descent from a very ancient common ancestor. This is possible. Ancient Siberian populations were not static, and the haplogroups present in Siberia 20,000 years ago may have been quite different from those found there today. But the alternative explanation, that haplogroup X in North American populations reflects an actual ancient contact event across the North Atlantic by populations that carried this haplogroup at higher frequencies, has been argued with genuine scientific rigor by researchers who are not fringe theorists. The Solutrean populations of Ice Age Europe and the Basque region were among the populations where haplogroup X was present. If any contact occurred, the haplogroup would have travelled with the people. And the geographic distribution of haplogroup X in North America, concentrated in the northeastern region and particularly around the Great Lakes, is exactly what you would expect if people arriving from the North Atlantic had mixed with local populations along the eastern seaboard and around the Great Lakes, before that mixed population dispersed further inland. This does not prove anything. The conventional explanation remains viable. But it means that the Rh negative question and the haplogroup X question are pointing in the same direction, toward the same region of North America, suggesting the same kind of ancient Atlantic contact and producing the same level of scientific uncertainty, combined with suggestive but not conclusive evidence. There are stories told among the Anishinaabe peoples, a broad cultural group that includes the Ojibwe, the Odawa, and the Potawatomi, who have inhabited the Great Lakes region for as long as oral history can reach, about people who came from the east across the Great Water and were different in appearance from the Anishinaabe themselves. These stories have various versions and various interpretations, and the specific details differ between communities and storytellers. But the common thread is the arrival of a people, sometimes described as light-skinned or with unusual physical features, who came by boat from across the sea and who interacted with the people of the Great Lakes region in a variety of ways. Some versions of the story describe these visitors as bringing gifts and trading peacefully. Some describe conflict. Some describe intermarriage and the absorption of the newcomers into existing communities. These oral traditions have been largely dismissed by Western historians as mythological accounts that do not describe actual historical events. The argument is that ancient peoples, like all peoples everywhere, created stories to explain the unknown, and that the arrival of strangers from across the sea is a common mythological theme that appears in oral traditions from many cultures without necessarily reflecting a literal historical memory. This dismissal may be correct, or it may be a mistake born of the systematic undervaluing of indigenous knowledge systems that has characterized Western scholarship for centuries. The problem with dismissing oral traditions as mere mythology is that there are now well-documented cases where oral traditions have been shown to preserve accurate historical information across thousands of years. Researchers have found cases where Aboriginal, Australian oral traditions appear to accurately describe the flooding of coastal sites that occurred more than 7,000 years ago at the end of the last ice age. They have found cases where Mi'kmaq oral traditions about specific locations correlate with archaeological discoveries at those same locations. The accuracy of oral traditions as historical records is not unlimited, and not every oral tradition is a reliable account of a specific historical event. But the categorical dismissal of indigenous historical memory as irrelevant to actual history is a position that serious researchers have increasingly abandoned. What the Anishinaabe traditions say about strangers from the East fits remarkably well with what the genetic and archaeological evidence suggests about the possible ancient history of the Great Lakes region. It does not prove anything, but it adds another thread to a web of evidence that, when you pull it taut from multiple directions, begins to show the outline of something real. Now let us move from oral traditions to physical evidence of a more unusual kind. Because one of the most persistent and puzzling features of North American archaeology is the occasional discovery of human remains that do not fit the standard profile of Northeast Asian-derived ancestry. These discoveries are controversial, often fiercely so, and the controversy is not purely academic. It involves questions of indigenous sovereignty, cultural patrimony, and the political dimensions of claiming that some ancient North Americans were of non-Native American ancestry. These are real and serious concerns that need to be acknowledged. But the scientific questions they touch on are also real, and the evidence that has accumulated over decades of research is not nothing. The most famous of these cases is Kennewick Man, a set of skeletal remains found along the Columbia River in Washington state in 1996. Kennewick Man was a male individual who died approximately 8,500 years ago, making him one of the oldest known individuals in North America. His skeletal morphology, meaning the shape and proportions of his skull and body, was analyzed by physical anthropologists and found to be unusual. It was not typically Native American by the standards of modern populations. It showed features that some researchers described as more similar to certain Southeast Asian populations, or to the ancient Jomon people of Japan, or in the most contentious interpretations, to early European populations. The Kennewick Man case became one of the most legally and scientifically contested archaeological discoveries in American history. Native American tribes claimed repatriation rights under federal law. Scientists argued that the remains were too old to be culturally affiliated with any existing tribe and should be available for study. After years of legal battles, the remains were eventually returned to a coalition of tribes for reburial. But before that happened, significant scientific analysis had been conducted, and the results were genuinely puzzling. When ancient DNA was finally extracted from Kennewick Man's remains and analyzed, the results showed that he was indeed most closely related to modern Native American populations, resolving the question of whether he was Asian or European in origin in favor of the American side of the argument. But the ancient DNA also showed genetic features that were not typical of modern Native American populations, suggesting that the founding populations of the Americas were more diverse than the current distribution of indigenous people would suggest. The diversity that exists in the oldest remains from the Americas is not fully preserved in modern Native American populations, suggesting that population replacement events, bottlenecks, and the massive mortality of the post-Columbian period all contributed to simplifying the genetic picture. This means that the genetic diversity of ancient North Americans, including whatever variation existed in blood type frequencies and in haplogroup distributions, was greater than we can directly infer from modern populations. The ancestors of modern Native Americans were not a genetically uniform group. They were descended from multiple migration events, possibly multiple contact events with non-Siberian populations, and the genetic complexity that would have existed in ancient North America was in many ways larger and stranger than what has survived into the modern era. Canada is at the center of this question geographically. The eastern coastline of Canada was among the first places in the Americas to be reached by any Atlantic crossers, whether those were Ice Age Solitarian people 20,000 years ago, Norse explorers 1,000 years ago, or Basque fishermen 500 years ago. If any of those contacts left a genetic trace, that trace would have entered the gene pool of northeastern North America and would have had thousands of years. in the case of the earliest contacts, to spread through indigenous populations via the trade networks and seasonal movements that connected communities across the continent. The question of how far such a genetic signal could spread in any given time frame is a mathematical one, with a reasonably clear answer. Gene flow through human populations is not instantaneous, but it is also not negligible. In a network of interconnected communities where trade and intermarriage occur at even modest rates, a new allele introduced at one point can spread across a very large geographic area within a few thousand years. The old copper trade networks alone, which we know connected the Great Lakes to communities from the Gulf Coast to the Atlantic seaboard, would have been more than sufficient to distribute a new genetic variant from its point of introduction into the broader North American gene pool within the time frame we are considering. So the picture that emerges from all of this, if you step back far enough to see it whole, is not one clean story with a clear beginning and a definitive explanation. It is a palimpsest. A document that has been written on, partially erased, and written over again multiple times, so that fragments of older writing show through under the newer layers if you look carefully enough and at the right angle. Canada's Rh negative frequencies are the latest layer of ink. Underneath them, if you look carefully, you can see traces of French colonization and Basque whaling and Norse exploration. And underneath those, if you look harder, you can see the faint outlines of something older still, ancient haplogroups. Trade networks built around copper that nobody can fully account for, oral traditions describing strangers from the East, skeletal morphologies that refuse to fit neatly into any established category, and a blood type marker that appears, in certain communities and certain regions, at frequencies that no one explanation, applied alone, can fully account for. Let us talk now about Atlantic Canada specifically, because the provinces of Nova Scotia, New Brunswick, Prince Edward Island, and Newfoundland occupy a geographic position that makes them the most likely entry point for any Atlantic contact event, and they present a cluster of anomalies that is particularly dense and particularly compelling. Nova Scotia was called Acadia by the French colonists, who settled there in the early 1600s, and the Acadians who developed there before being brutally expelled by the British in the 1750s are one of the most genetically studied populations in Canada. The Acadian founding population was relatively small, arrived over a relatively short period, and experienced significant genetic drift due to geographic isolation and the founding population effect. Genetic studies of Acadian descendants have found elevated Rh negative frequencies that are consistent with an origin population that included a significant Basque component, which historical records confirm, since many of the earliest Acadian settlers came from regions of France with strong Basque cultural and commercial connections. But what the genetic studies have also found, in some cases, are markers that appear to reflect a degree of indigenous admixture in the earliest Acadian settlers that is higher than the historical record explicitly acknowledges. The early Acadian colonists interacted extensively with the Mi'kmaq. Some historical records document marriages between French settlers and Mi'kmaq women. The proportion of Mi'kmaq ancestry in modern Acadian descendants is a matter of ongoing research, and the estimates vary, but few serious researchers believe it is zero. And, as we have discussed, the Mi'kmaq themselves carry genetic markers that are unusual, and that may reflect ancient contact events going back well before European colonization. So in the Acadian population, you potentially have multiple layers of Rh negative input overlaid on each other. The Basque contribution to the founding French origin settlers, whatever pre-Columbian contact had deposited in the Mi'kmaq gene pool over centuries or millennia. And then, on top of that, the mixing of those two sources in the early colonial period. Disentangling these layers is not straightforward, and the research that would be needed to do it properly, specifically ancient DNA analysis from pre-contact Mi'kmaq remains, is complicated by issues of indigenous sovereignty and community consent that are not going to be resolved quickly or easily. Newfoundland is a special case within Atlantic Canada because of its documented Norse connection. The L'Anse aux Meadows site is only the beginning of the story. The Norse sagas describe multiple voyages to areas they called Helluland, Markland, and Vinland, which most researchers identify with Baffin Island, Labrador, and Newfoundland, or the Gulf of Street, Lawrence, respectively. The contacts described in the sagas are not purely peaceful or purely brief. One saga, the Saga of Eric the Red, describes a Norse woman named Freydis in violent and extended conflict with Skraelings, the Norse term for indigenous people. Another saga, the Saga of the Greenlanders, describes the Norse buying furs from Skraelings in exchange for red cloth. These are not descriptions of brief, incidental encounters. These are descriptions of sustained commercial and military interactions with indigenous populations. The genetic trace of the Norse in indigenous Newfoundland populations has been searched for and has so far not been found in any definitive, unambiguous way. This is partly because of the difficulty of distinguishing Norse genetic contributions from later European admixture in modern populations, and partly because of the limited ancient DNA available from the relevant period. But absence of proof is not proof of absence, and the sagas describe conditions, sustained contact, trade relationships, possibly intermarriage or sexual contact of various kinds, under which gene flow would have been almost inevitable. The Norse of the year 1000 CE were largely of Scandinavian origin, with some admixture from their earlier contacts with Celtic populations in Ireland and Scotland. They carried Rh-negative frequencies in the range of 15 to 20%, consistent with Northern European norms. If any Norse individuals remained in North America or had children with local women, those children would have had a meaningful probability of being Rh-negative. Over several hundred years of occasional contact, which is what the sagas and the archaeological evidence together suggest, the cumulative genetic contribution to local populations could have been small but non-trivial. And there is one more dimension to the Newfoundland story that brings us to one of the most poignant and mysterious aspects of Canadian pre-contact history: the Beothuk. The Beothuk were the indigenous people of the island of Newfoundland at the time of European contact. They are now extinct as a distinct people. The last known member of the Beothuk, a woman named Shanordithit, died in 1829. She left behind drawings and accounts that are among the most precious records of indigenous Newfoundland culture, but the Beothuk culture and language died with her, and the genetic profile of the Beothuk died with her population before anyone thought to study it systematically. What we know of the Beothuk from historical accounts and archaeological evidence is suggestive rather than definitive. They were described by early European observers as having reddish or ochre-coloured skin, which is almost certainly a reference to the practice of coating their bodies with red ochre, a pigment they used extensively in life and in burial, similar to practices documented in ancient burial sites across North America and in Neanderthal contexts in Europe. Whether the Beothuk's use of red ochre reflects a cultural continuity stretching back to an ancient contact with people who carried this practice from Europe is unknowable at this point. But it is another thread in the web of suggestive connections that link Atlantic Canada to ancient Atlantic crossings that no historical document has recorded. The red ochre connection is worth pausing on because it keeps appearing across the most interesting and most puzzling archaeological sites in North America, and because the specific way it appears in certain contexts suggests something that goes beyond simple coincidence of independent cultural development. Red ochre is found in human burials going back hundreds of thousands of years on multiple continents. It is one of the most ancient forms of symbolic behavior that we can trace archaeologically, and its widespread occurrence is usually attributed to its symbolic resonance with blood and life and the red of the rising and setting sun. It is a universal human symbolic language expressed through pigment, and the fact that it appears in burial practices on both sides of the Atlantic does not by itself prove anything about contact between those two traditions. But the specific way red ochre is used in certain ancient North American burial traditions goes beyond simple symbolic gesture into something that looks more like a systematic cultural practice with direct parallels to Old World traditions. The maritime archaic culture, which developed along the Atlantic coast of Canada and the northeastern United States beginning roughly 8,000 years ago and persisting for more than 5,000 years, used red ochre in their burials in quantities and patterns that are remarkable. They stained the bones of the dead with ochre, covered grave goods with ochre, lined burial pits with ochre. This was not a casual use of a symbolic pigment. This was a systematic, extensive, culturally significant practice that required substantial effort and reflected a specific theological or cosmological framework. And the maritime archaic culture is one of the most archaeologically interesting traditions in all of North American prehistory, not only because of the red ochre, but because of the sheer sophistication and ambition of the culture it represents. These were not simple hunter-gatherers in the popular imagination of that term. They were maritime specialists who had developed technologies for hunting large sea mammals, including whales and walruses, in the open North Atlantic. They were using harpoons with toggling tips, a technology that creates a secure hold on a large marine animal by pivoting at the point of impact, that was previously thought to have been a much later development. They were building seaworthy watercraft capable of operating in the open ocean, not just coastal waters. And they were maintaining trade networks and cultural connections across an enormous geographic range, from the Gulf of St. Lawrence to Labrador to Newfoundland. The maritime archaic people were the dominant cultural tradition in Atlantic Canada for thousands of years. They are among the ancestors of the historical indigenous populations of the region. And when their cultural remains are examined alongside the oral traditions of the Mi'kmaq and other Algonquian peoples, certain connections emerge that suggest a deep continuity stretching from the maritime archaic period all the way into the documented historical era. What was the genetic profile of the maritime archaic people? This is another question that we cannot fully answer because of the poor preservation of ancient DNA in the acidic soils of the Atlantic Canadian coast. The few samples that have been analysed are tantalising but incomplete. Some researchers have noted features of the available ancient DNA that are consistent with a population that was genetically distinct from later indigenous populations of the region, possibly reflecting an earlier migration event or a founding population that was subsequently partially replaced by later incoming groups. The question of whether the maritime archaic people carried unusual blood type frequencies, including elevated Rh negative, is not one that the current data can answer. But the question is worth asking, and it is being asked quietly in genetics labs where the full implications of the answer are understood. Let us now push the timeline even further back and consider a possibility that will sound extreme but is grounded in specific and serious research. There is evidence, contested but substantial, that humans were present in North America significantly earlier than the conventional timeline of 15,000 to 20,000 years ago suggests. The site of Bluefish Caves in Yukon has produced animal bones with cut marks that have been dated to as much as 24,000 years ago. The site of Monteverde in Chile has been radiocarbon dated to approximately 14,500 years ago, which requires that the people who made it had been in the Americas for several thousand years before that. The site of Cerruti Mastodon in California produced mastodon bones with evidence of human modification that has been dated, controversially, to approximately 130,000 years ago, a date so early that it would require a complete rethinking of human prehistory, not just North American prehistory. The Cerruti Mastodon date is so extreme that most researchers treat it with great skepticism, and there are legitimate questions about whether the bone modifications were actually made by humans or could have been produced by geological processes. But the Bluefish Caves dates and the Monteverde date are within the range of serious scientific debate, and they suggest that the Americas were first inhabited earlier than the standard model implies. If humans were in the Americas 24,000 years ago, they were there during the last glacial maximum, when the Beringia land bridge was at its widest and the ice-free corridor through Canada was not yet open. The only viable route for human migration at that time was a coastal route along the edge of the Pacific, or, far more controversially, a coastal route along the edge of the North Atlantic ice sheet. A coastal route along the North Atlantic ice edge during the last glacial maximum is exactly the scenario that the Solutrean hypothesis proposes. And if that route was viable, then the people who took it were not Asian in origin, they were European. They came from a continent where haplogroup X was present, where Rh negative blood was carried at frequencies far higher than in any East Asian population. And they arrived in North America at a time when the continent was essentially uninhabited, which means they were not a small trickle of migrants mixing into an existing large population. They were potential founders, or co-founders, of the North American gene pool. The implications of this for the Rh negative question are obvious. If the Solutrean hypothesis is correct, even partially, even in the sense that some Atlantic crossing occurred at some point in the deep past, before the Norse or the Basque, then the Rh negative marker in North American populations is not merely a European colonial artifact. It is part of the founding genetic signature of this continent, carried by some of the first people who ever set foot on it, and preserved in diluted but detectable form in some of the populations that descend from them today. This is where some researchers stop and some researchers keep going. The careful ones stop here and say that while the hypothesis is interesting and the evidence is suggestive, it is not proven, and extraordinary claims require extraordinary evidence, and the evidence for the Solutrean hypothesis has not yet cleared that bar. The less careful ones, and there are quite a few of them online and in the popular literature, take the suggestive evidence and use it to build grand narratives about ancient European civilizations that predate the mainstream historical record, about secret knowledge suppressed by academic establishments, about bloodlines that carry special powers or special destinies? This is where the legitimate scientific question gets hijacked by the human need for grand narratives, and the result is a confusion between what the evidence actually suggests and what people want it to mean. What the evidence actually suggests, at its most careful and rigorous, is this. Canada has Rh-negative blood frequencies that are higher than a simple model of recent European immigration would predict, particularly in certain specific regional populations and certain specific communities with indigenous ancestry. The explanation for these frequencies is almost certainly layered, involving recent European immigration as the dominant signal, but potentially also involving much older contact events, whether Norse, pre-Norse European maritime, or very ancient Atlantic crossings that left earlier, smaller, but not entirely negligible genetic traces. The data does not resolve cleanly into any single explanation, and that ambiguity is itself the most important thing about it, because it tells us that the history of human movement into and within the Americas was more complex, more multidirectional, and more ancient than the standard model allows. The people who should care most about this question are Canadians themselves. Canada is a country that has been grappling seriously in recent decades with questions about its own history, about the relationship between the settler colonial population and the indigenous peoples who preceded them, about what was here before Europeans arrived and what was done to it. The genetic mystery of Rh-negative blood in Canada is connected to all of those questions, not as a distraction from them, but as an additional dimension of the same fundamental inquiry. Who were the people who were here before? What was the full extent and depth of their history? What connections did they have to the wider world? And what happened to that history when European colonization reorganized everything? The people of Atlantic Canada, the descendants of Acadian farmers and Mi'kmaq fishermen and Basque whalers and Norse explorers and maritime archaic hunters, all of them carry pieces of this puzzle in their DNA. Some of those pieces are well understood. Some of them are still being analyzed. And some of them are lost, dissolved into the sediment at the bottom of the Atlantic Ocean with the villages of the people who drowned when the seas rose 14,000 years ago. Let us think for a moment about the geography of the North Atlantic in a way that most people have never been asked to think about it. Take a globe or a map that shows the Arctic regions without the distortion of the standard Mercator projection, and look at the relationship between Western Europe and northeastern North America. From the northern tip of Scotland to Iceland is about 800 kilometers. From Iceland to Greenland is about 300 kilometers at the narrowest point. From Greenland to the northern tip of Baffin Island is about 300 kilometers. These distances are not trivial, but they are not the open ocean crossing that most people imagine when they think about crossing the Atlantic. This is the North Atlantic stepping stone route. It is the route that the Norse used, island by island, from Norway to the Faroe Islands to Iceland to Greenland to North America. It is the route that seabirds use. It is the route that marine mammals follow. And it is, critically, a route where the northern portions were covered by continuous sea ice during the last glacial maximum, which would have allowed anyone willing to travel on ice, and there are ancient peoples who certainly were, to walk from what is now Scotland to what is now Canada, without ever needing a boat at all. Just a willingness to cross frozen ocean, knowledge of how to hunt seal and walrus along the ice edge, and the extraordinary hardiness that Ice Age people who survived in the North Atlantic region had already demonstrated. The Dorset culture, which is not the same as the Thule culture that eventually became the Inuit, occupied the Arctic regions of Canada from roughly 800 BCE to about 1500 CE. The Dorset people were remarkable. They were not ancestral to any modern indigenous group. They were a distinct lineage that had developed over thousands of years in the high Arctic, and they were gone by the time European explorers started documenting the Arctic in any detail, replaced or displaced by the expanding Thule culture from the west. The Norse encountered them and called them Skraelings, which appears to be the same word they used for the indigenous people further south. The Norse accounts of their encounters with Dorset people, if that is indeed who the Skraelings of the northernmost sagas were, describe people who were terrifying in their strangeness, who used skin boats and had technologies adapted to the Arctic in ways the Norse could not replicate, and who ultimately proved unwilling to coexist peacefully with Norse attempts at settlement in the high Arctic. The Dorset people are biologically mysterious. The ancient DNA that has been extracted from Dorset remains shows a population that was genetically distinct from both modern Inuit and from the ancestral Native American populations to the south. They appear to have been descended from one of the earliest migrations into the Arctic, possibly a separate migration event from the one that produced most Native Americans. Their genetic signature does not map cleanly onto any existing population in the world, and their blood type frequencies, where they can be inferred from the genetic data, include features that are unusual by the standards of any known ancient North American population. Whether the Dorset people carried elevated Rh-negative frequencies is not something the current data can definitively answer. But the question is interesting because the Dorset inhabited exactly the geographic range that would have been most exposed to any contact with people crossing the North Atlantic from Europe, and they inhabited it for over 2,000 years before their disappearance. If any Europeans reached the high Arctic, they would have encountered the Dorset. And if any gene flow occurred as a result of those encounters, the Dorset population would have been the vehicle through which it entered the North American gene pool. The disappearance of the Dorset is one of the great unresolved mysteries of North American prehistory. They were not simply absorbed by the incoming tool. The genetic record shows very limited Dorset ancestry in modern Inuit populations, suggesting that the transition from Dorset to Thule dominance involves substantial population replacement rather than absorption. The Thule, expanding east from Alaska around 800 CE, appear to have almost completely displaced the Dorset within a relatively short period. What happened to the Dorset people in those final generations? Where did they go? A small population, already diminished by pressure from the expanding Thule, already isolated at the margins of the habitable world, may simply have declined to extinction. Or they may have found refuge further south, mixing with other indigenous populations and losing their distinct cultural and genetic identity in the process. If the Dorset carried any unusual genetic markers, including possible Rh-negative alleles from ancient Atlantic contact events, those markers would have been diluted and lost in the general indigenous population of northeastern Canada when the Dorset dispersed or disappeared. They would be present today only as tiny, nearly undetectable traces in populations that do not have any cultural memory of Dorset ancestry, because the cultural memory died when the Dorset culture died. This is the nature of the problem with deep pre-contact history in Canada. The evidence exists. It is embedded in the DNA of living Canadians and in the ancient DNA that can sometimes be extracted from well-preserved remains. But it is diluted, fragmented, and overlaid by centuries of subsequent population movement and mixing that obscure the signal of whatever was there at the beginning. Now let us turn to one of the most specific and concrete anomalies in the Canadian Rh, negative distribution, one that has been noted by researchers and that has not received anything like the attention it deserves in mainstream discussions of Canadian genetics. This involves the Haudenosaunee, known to most Canadians and Americans as the Iroquois, the confederacy of 6 nations, whose territory historically spanned the region from what is now upstate New York to southern Ontario and Quebec. The Haudenosaunee are one of the most politically sophisticated indigenous peoples in North American history. Their confederacy, which united the Mohawk, Oneida, Onondaga, Cayuga, and Seneca nations, and later incorporated the Tuscarora as a 6th nation, is sometimes cited as an influence on the democratic structure of the United States Constitution. Their oral traditions, their governance systems, and their cultural practices have been extensively documented and are among the best preserved of any North American indigenous tradition. The Haudenosaunee also present some genetic puzzles that are not fully resolved. Studies of Haudenosaunee genetic profiles have found a more complex picture than the standard model of a purely Northeast Asian-derived indigenous North American population would predict. There are haplogroup frequencies and specific genetic markers in Haudenosaunee communities that are unusual, and the interpretation of those features is contested. Some researchers attribute them to genetic drift in a population that experienced significant bottlenecks during the colonial period. Others point to the Haudenosaunee practice of adopting large numbers of captives from other nations, which would have continuously introduced new genetic variants into the Confederacy population. And a small number of researchers have pointed to features that may reflect very ancient contact with non-Siberian populations, though this interpretation is the most speculative. What is not speculative is the geographic position of the Haudenosaunee homeland. Their territory was at the intersection of multiple ancient trade networks, including the Great Lakes Copper Network, the trade routes connecting the Street, Lawrence Valley with the Atlantic coast, and the networks running north-south along the major river systems of eastern North America. If any ancient contact event deposited unusual genetic markers into the northeastern North American gene pool, the Haudenosaunee, sitting at the hub of the most important regional trade networks, would have been among the populations most likely to accumulate those markers through trade-driven intermarriage and alliance. The blood type data for Haudenosaunee communities is limited and not always publicly available, partly because of the community sovereignty over genetic research that most First Nations have appropriately asserted. What is available suggests frequencies that are somewhat higher than the East Asian baseline, but lower than the surrounding Canadian population. The degree to which this reflects ancient genetic complexity versus recent European admixture is unclear. The Mohawk Nation, which has substantial communities on both sides of the Canada-United States border, has been particularly resistant to genetic research that it has not initiated and controlled for very understandable historical reasons. The history of scientific research on indigenous bodies and biological materials in North America is a history of exploitation and disrespect that justifies significant caution. This means that some of the most important data for understanding the Rh-negative question in Canada is not currently available to researchers, and will not be unless Indigenous communities decide that the research serves their interests and conduct it on their own terms. This is as it should be. The questions about pre-contact Canadian history belong in the first instance to the people whose ancestors made that history, and the answers will come, if they come at all, on a timeline and under conditions determined by those communities. In the meantime, researchers have been working with the publicly available genetic data and with the archaeological record to try to build the best picture they can of what happened in Canada before the documented historical era. And one of the most interesting lines of inquiry has been the investigation of pre-contact transoceanic contact, not from the direction of Europe but from the direction of Polynesia. There is now solid genetic evidence, published in rigorous peer-reviewed journals, that Polynesian people made contact with the indigenous inhabitants of South America sometime around 800 CE, more than 600 years before Columbus. This evidence comes from the genetic analysis of modern indigenous South American populations, which carry a small but unmistakable Polynesian genetic component, and from the archaeological evidence of the sweet potato, a plant that originated in South America and was distributed across Polynesia well before European contact. The Polynesians reached South America. They traded with the people there, or they were received by the people there, and some degree of genetic exchange occurred. This is not relevant to Canada directly, because the Polynesian contact with South America occurred far to the south of any Canadian territory, but it is relevant in the broader sense that it demonstrates that pre-Columbian transoceanic contact between the Americas and the rest of the world was real, that it occurred more than once, from more than one direction, and that the genetic evidence of such contact, while small, is detectable and analyzable with modern techniques. If Polynesian contact with South America left a detectable genetic trace, then European contact with North America at an earlier date could have left a detectable genetic trace as well, and that trace might include Rh-negative alleles. The question is whether the Rh-negative frequencies we see in Canada today represent a trace signal of ancient contact events overlaid by the dominant signal of recent European immigration, or whether the recent European immigration is the entire explanation and the ancient contact hypothesis is a compelling story built on ambiguous data. This is a genuinely open scientific question, and the people best positioned to answer it, population geneticists, with access to large datasets of ancient and modern DNA from Canadian populations, are working on it right now. In universities from Halifax to Vancouver, the answers, when they come, will not be dramatic press releases announcing that ancient Europeans crossed the Atlantic and mixed with Native Americans. They will be careful papers, published in genetics journals, full of statistical confidence intervals and caveats, slowly shifting the scientific consensus in one direction or another, but science rarely tells the full story. The statistical confidence intervals do not capture what it meant to be a Basque sailor who spent 3 winters on the Labrador coast before the fishing season, learning enough of the local language to trade, and leaving a child behind when the ships finally sailed home. They do not capture what it meant to be a Mi'kmaq woman who lived with a Norse traveller for a year and a half and watched him sail away toward the horizon, and who carried his child to term and raised that child among her own people, and passed on something in the blood that would travel forward through 20 or 30 generations until it ended up in a tube of blood drawn in a Halifax hospital in the 21st century, slightly too Rh-negative to be fully explained by the demographic history that the textbooks record. That is the human story underneath the statistics. It is not a story of invasion or conquest or displacement, at least not at the level we are discussing here. It is a story of contact, of exchange, of the ordinary human things that happen when people from different places meet each other at the edge of known worlds. Trade, curiosity, desire, children, and blood. There is another dimension of this mystery that most discussions of Rh-negative blood in Canada entirely overlook, and it involves not the origin of the blood type, but its persistence. Even if we accept that European immigration introduced elevated Rh-negative frequencies into the Canadian population over the past 4 centuries, that acceptance still leaves open the question of why those frequencies have remained as high as they are, and in some communities, appear to be higher than what simple demographic mixing would predict. This is a question about selection. Natural selection works on genetic variants by favoring the carriers of beneficial variants and disfavoring the carriers of harmful ones. For blood type variants, the most significant selective pressure is the one that the Rh system creates through pregnancy complications. An Rh-negative woman carrying an Rh-positive baby faces an immune challenge that, before modern medicine, sometimes resulted in the death of subsequent Rh-positive babies. This is a real selective pressure, and it works against the Rh-negative variant in mixed populations. All else being equal, in a population where Rh-negative women regularly have Rh-positive babies, the frequency of the Rh-negative allele should decline over time. The fact that Rh-negative frequencies remain as high as they are in Canada, across multiple generations, and under conditions where intermarriage between Rh-positive and Rh-negative individuals has been common, suggests one of two things. Either there is a countervailing selective advantage associated with being Rh-negative that offsets the reproductive disadvantage, or the frequency is being maintained at something close to a demographic equilibrium by immigration from Rh-negative rich source populations. Both explanations are probably partially correct, but the first one is the more interesting. The hypothesis of a selective advantage for Rh-negative individuals is not new, but it has gained more traction in recent decades, as the tools for studying immune system genetics have become more sophisticated. The basic idea is that the same immune system characteristics that make Rh-negative mothers vulnerable to sensitization against Rh-positive fetuses might also give them advantages in fighting off certain pathogens. An immune system that is particularly sensitive and reactive, that creates antibodies quickly and maintains them strongly, is a system that is better at recognizing and destroying foreign biological material in general. The cost of that sensitivity, in the form of occasional attacks on Rh-positive fetuses, might have been offset in ancient environments by the benefit of superior resistance to certain infectious diseases. Which diseases? This is where the research is still thin and the conclusions are still tentative. Some studies have found that Rh-negative individuals show different rates of infection with certain pathogens compared to Rh-positive individuals, but the results are inconsistent across studies and populations, and no single pathogen has been identified as the clear driver of Rh-negative selection. The most promising candidates involve parasites rather than viruses, because parasites tend to exert stronger and more consistent selective pressure on immune system genes. But the specific mechanism remains unclear. There is another hypothesis, one that is speculative but intriguing, which connects the selective maintenance of Rh-negative frequencies to the specific demographic and epidemiological history of Canada. The ancestors of modern Canadians, both Indigenous and European origin, lived through a series of catastrophic disease events in the 17th, 18th, and 19th centuries that radically reduced Indigenous populations and reshaped the genetic landscape of the continent. If Rh-negative individuals were even slightly more resistant to any of the epidemic diseases that swept through North America during the colonial period, smallpox being the most devastating but by no means the only one, then the post-epidemic survivors would have included a disproportionately high proportion of Rh-negative individuals. Their descendants would carry the Rh-negative allele at higher frequencies than the pre-epidemic population had. This is a dark hypothesis. It would mean that the elevated Rh-negative frequencies we see in Canada today are partly a legacy of genocide, a biological echo of the mass death that colonial disease brought to Indigenous populations and that reshaped the genetic landscape of the entire continent. Whether it is true cannot be determined from current data. The ancient DNA from pre-epidemic Indigenous populations is not complete enough or large enough to compare pre- and post-epidemic Rh-negative frequencies with any statistical confidence. But it is a hypothesis that sits uneasily in the space between epidemiology and history, reminding us that genetic patterns are never politically neutral. They always have a history, and that history is always entangled with power. Let us step back from the dark history for a moment and look at the simple geography of the Rh negative distribution in Canada through a different lens. Instead of asking where it came from, let us ask where it is now and what that tells us about how it has moved through the Canadian population over time. The highest concentrations of Rh negative blood in Canada are found in the provinces with the deepest European settlement histories. Quebec and the Atlantic provinces show the highest frequencies, reflecting the large founding populations of French and British settlers who began arriving in the early 1600s. These founding populations came disproportionately from regions of Europe with high Rh negative frequencies, the Basque-influenced areas of southwestern France for the French Canadians, and Scotland and Ireland for many of the British and later Acadian-descended settlers in the Atlantic provinces. Scotland carries Rh negative frequencies above 20%. Ireland is similar. The Scottish Highlands, where the original Gaelic-speaking population has been most continuously preserved, shows frequencies that rival some of the highest in Europe. The Scottish connection to Canada is worth examining more closely, because the Scots who came to Canada were not a random sample of the European population. The Highland clearances of the late 18th and early 19th centuries drove enormous numbers of Scottish Highlanders out of their ancestral territories in the northern and western parts of Scotland and onto ships bound primarily for Nova Scotia, Prince Edward Island, and Ontario. These were people from communities that had been relatively isolated for centuries, that had experienced the genetic drift and founder effects of small, bounded highland communities, and that carried genetic profiles reflecting thousands of years of continuity in one place. Among those genetic profiles were the haplogroups and blood type frequencies of the ancient Celtic-influenced populations of northwestern Europe, including haplogroup X, which appears in elevated frequencies in some Scottish Highland communities compared to the broader European average. The Irish who came to Canada, primarily during and after the Great Famine of the 1840s, represented a somewhat different genetic profile. The Irish population, particularly the rural Western Irish population that produced the bulk of the famine immigrants, had also experienced long periods of relative isolation and had maintained genetic features associated with the ancient Atlantic coastal population of Western Europe. The Western Irish show some of the highest Rh negative frequencies in the British Isles, and the descendants of famine Irish settlers in cities like Montreal, Toronto, and Halifax carry those frequencies forward. So in the Atlantic, Canadian, and Quebec populations, you have a mixture of founding European groups, all drawn from the Western European populations, with the highest Rh negative frequencies on the continent, layered over whatever indigenous ancestry existed in those populations, and then further mixed with the continuous immigration that Canada has experienced throughout its history as a destination country. The result is a population with Rh negative frequencies that are genuinely elevated compared to the global average, and that reflect the specific demographic history of a country built primarily on Atlantic crossings. But Canada is not only its Atlantic coast. The prairie provinces, the Great Plains of Manitoba, Saskatchewan, and Alberta, have a different genetic history. The indigenous peoples of the prairies, the Cree, the Blackfoot, the Assiniboine, the Plains, Ojibwe, and many others, had contact with European settlers primarily from the mid-19th century onward, somewhat later than the contact events that shaped the Atlantic provinces. The European settlers who moved onto the prairies were more ethnically diverse than the founding populations of the East, including not only British and French, but also large numbers of Ukrainians, Germans, Scandinavians, Poles, and other Central and Eastern European groups who came to farm the new land in the late 19th and early 20th centuries. The Rh negative frequencies among these Central and Eastern European populations are lower than those of the Atlantic European groups, because Central and Eastern Europe has historically lower Rh negative frequencies than Western Europe. As these populations mixed on the Canadian prairies, the resulting frequencies reflect that mixture, somewhat lower than in the East. And the indigenous contribution to prairie genetic profiles, where it exists in mixed indigenous and European ancestry communities, is complicated by the specific histories of those communities and the degree to which they have preserved or lost ancestral genetic patterns in the generations since contact. The Northern Territories, the Yukon, the Northwest Territories, and Nunavut represent the most complex genetic picture of all. The indigenous peoples of the far north, the Dene Nations and the Inuit, have genetic profiles that are more purely derived from the ancient Arctic and subarctic migrations than any other Canadian indigenous group. The Inuit in particular show genetic profiles very close to the Siberian and Yupik populations from whom they descend, and Rh-negative frequencies in traditional Inuit communities are very low, consistent with the East Asian baseline. The Dene nations of the Yukon and Northwest Territories show somewhat more complex profiles, reflecting both their ancient ancestry and the various contact events, with other indigenous groups, with European fur traders, and with the particular demographic history of the northern fur trade economy. The far north is also the region where the archaeological evidence is sometimes best preserved, because the permafrost that covers much of it can protect organic material for thousands of years in a way that the temperate soils of the south cannot. Some of the most important ancient DNA from Canada has come from permafrost-preserved remains in the north, and while most of what has been extracted and analysed is consistent with the standard model of indigenous North American genetics, there are occasional outliers, specimens whose genetic profiles do not fit neatly into any established category, that keep the questions open. One of these outliers was found in the Yukon, at a site called Spirit Cave. The Spirit Cave mummy, found in Nevada rather than Canada, but representative of the kind of anomalous ancient remains that appear periodically across the continent, showed a genetic profile that puzzled researchers when it was first analysed. More recent analysis of similar anomalous remains from the broader region has gradually built a picture of the Ice Age and early post-Ice Age Americas as genetically more diverse than the modern indigenous population would suggest. Multiple founding lineages, multiple migration events, possibly, in the ancient populations of the eastern portion of the continent, some input from directions other than Siberia. The mystery deepens when you start looking at specific archaeological sites in Canada that have produced anomalous finds, objects, or evidence that does not fit the expected pattern of the time and place in which they were found. Some of these finds are well documented and legitimate. Others have been misidentified, misinterpreted, or in some cases, fabricated by people with particular agendas. Sorting the real anomalies from the false ones is one of the most demanding tasks in alternative archaeology, because the field attracts both serious researchers and determined fantasists in roughly equal measure. Among the legitimate anomalies, one of the most compelling is a site in Nova Scotia called Debert. The Debert site is one of the oldest confirmed human habitation sites in Atlantic Canada, with radiocarbon dates placing its occupation at roughly 10,600 years ago. The people who lived at Debert were Paleo-Indians, members of the earliest human populations to inhabit northeastern North America after the glaciers retreated. Their tools belonged to the Clovis tradition, and they were hunting now extinct megafauna, including caribou and possibly mastodon, in a landscape that had only recently emerged from beneath the ice. What makes Debert interesting beyond its antiquity is the evidence it provides for how quickly the post-glacial landscape was being colonized by human populations. Within a few thousand years of the ice retreat, people were living at the far eastern edge of the continent, at the very doorstep of the North Atlantic. They were not simply following the ice edge southward from Siberia. They were already at the geographic point where, if any transatlantic contact were to occur, it would most likely occur first. The connection between Debert and the Rh-negative question is indirect. We have no genetic data from the Debert people. Their remains, such as they are, have not yielded ancient DNA. But they are among the ancestors of all subsequent human populations in Atlantic Canada, and whatever genetic profile they carried was part of the founding genetic stock of northeastern North American indigenous people. If they carried any unusual genetic markers, those markers were available to be passed forward to later generations. From Debert, the human presence in Atlantic Canada expanded and diversified over thousands of years. The maritime archaic culture emerged from these Paleo-Indian roots. The woodland cultures that followed the maritime archaic brought new technologies, including pottery, agriculture in some regions, and the elaboration of burial traditions that show continuity with ancient practices going back to the earliest Paleo-Indians. All of these populations were in contact with each other, trading, migrating, marrying across group boundaries, and exchanging the genetic information that over time produced the indigenous peoples who were present when European contact began. This long human history, 12,000 or more years of continuous population growth, diversification, and exchange in what is now Canada, is the essential context for the Rh-negative question. When you ask where Canada's Rh-negative blood came from, you are not asking a simple question with a simple answer. You are asking about the full depth of human history on this continent, about the complexity of ancient migration and contact, about the ways in which populations that seem separate and distinct have always been more connected than they appear. The answer that most scientists would give you today is still primarily the European immigration answer. Yes, Canada's Rh-negative frequencies are high because a large proportion of Canada's population is of Western European ancestry, drawn from exactly the populations that carry Rh-negative blood at the highest frequencies in the world. This is true. This is real. This accounts for the majority of the signal. But the majority is not the whole story. And the parts of the story that the European immigration explanation does not capture are the parts that make a geneticist or an archaeologist or a historian sit up straight and pay attention. Because those parts are pointing towards something older, something that happened before the ships of Columbus or Cabot or Champlain crossed the Atlantic, something that left a trace in the blood of people who have been on this continent for thousands of years, and that science is only now beginning to have the tools to properly investigate. The tools are getting better very quickly. Ancient DNA analysis, which was barely possible 30 years ago and was limited to a handful of samples 10 years ago, is now being applied to hundreds and thousands of specimens across North America. The databases of ancient genetic data are growing every year. The methods for detecting and characterizing small genetic signals from ancient contact events are becoming more sensitive and more accurate. The questions that are currently answered with "we do not know" or "the data is insufficient" will have better answers within a decade. And when those answers come, they will change the story of Canada in ways that no one can fully predict right now. They may confirm that European immigration is the complete explanation and that the apparent anomalies in the data are statistical noise. They may reveal that pre-Columbian contact events, whether Norse, Basque, or something more ancient, left genetic traces that are small but real. They may find evidence of migration events and contact events that no current hypothesis has predicted, because the human past is always more complex and more surprising than any model can fully capture. Whatever the answers are, they belong to Canada. They belong to the people who live there, both the descendants of European settlers and the indigenous people whose ancestors were here for thousands of years before those settlers arrived. The story of human genetics in Canada is not a story about any single group's claim to the land or any hierarchy of belonging. It is a story about the extraordinary complexity of human movement and mixture across one of the world's most important geographic crossroads. It is a story about what blood carries and what blood means, in the literal biochemical sense, and in the deeper human sense of identity and belonging, and the question of where we come from. For the person sitting in a blood donation clinic in Halifax or Thunder Bay or Victoria, watching the needle draw their Rh-negative blood into a tube and wondering briefly what that label means, the answer is more complicated and more interesting than any label can capture. That blood carries the genetic signature of every person who contributed to the lineage that produced the individual sitting in that chair. It carries the signal of ancient migrations from Africa through Asia and into the Americas, and possibly the signal of other migrations that crossed other oceans in other directions. It carries the imprint of every population bottleneck, every epidemic, every founding event that shaped the communities their ancestors belonged to. It carries, possibly, traces so old and so faint that no current technology can detect them, of people who crossed ice or ocean when the world looked different from any map we recognize, and who left only the faintest fingerprint of their passage in the genes of the people who came after. That is what makes blood mysterious. Not its color or its chemistry, though those are remarkable in their own right. What makes blood mysterious is its depth. It goes back further than any memory. It carries information that no history book has recorded. And in Canada, where the human story is both very long and very incompletely understood, that mystery runs particularly deep. Let us now consider what the current frontier of research actually looks like, because the question of Rh-negative blood in Canada is not purely historical. It is being actively investigated in ways that will produce real scientific results within the coming years. And understanding how that research is being conducted helps clarify both what we might learn and what we cannot learn. The most important tool currently being applied to these questions is ancient DNA analysis combined with large-scale genomic data from modern populations. Researchers at institutions including the University of Toronto, McGill University, and several universities in the United States and Europe are building datasets that combine ancient DNA from archaeological specimens with modern genetic data from Indigenous and non-Indigenous Canadian populations. The goal is to reconstruct the genetic history of Canada with sufficient resolution to identify not just the major migration events, but the smaller, subtler gene flow events that followed them. The technical challenges are substantial. Ancient DNA from the warm and acidic soils of southern Canada is often poorly preserved, with DNA molecules broken into short, highly degraded fragments that are difficult to sequence and even more difficult to interpret. The permafrost sites in the north preserve DNA better, but the populations those sites represent are geographically distant from the regions where the Rh-negative anomaly is most pronounced. Extracting usable ancient DNA from the specific populations and the specific time periods most relevant to the question is extremely challenging, and in many cases the specimens simply do not exist because they were never excavated or have not been well preserved. The analysis of modern genetic data presents different challenges. The population structure of Canada today is complex, with recent immigration from dozens of different countries having added layers of genetic diversity on top of the older settler, colonial, and indigenous layers. Controlling for this recent diversity to extract information about older patterns requires sophisticated statistical methods that are only recently becoming available, and the portion of the Canadian population with significant indigenous ancestry, which is the portion most relevant to questions about pre-Columbian contact events, is also the portion most sensitive to the history of scientific exploitation that makes participation in genetic research a fraught proposition for many Indigenous communities. Despite these challenges, progress is being made. A study published in 2018 analyzed ancient DNA from individuals associated with the Clovis culture and early post-Clovis populations of North America and found evidence for the genetic diversity of the founding American population that is not preserved in modern Indigenous people. This diversity included components that were not present in known Siberian populations, consistent with either very ancient West Eurasian input via Siberia or with contact events that have not yet been fully characterized. The authors were careful about their conclusions, but they acknowledged that the founding genetics of the Americas are still not fully understood, and that the data is consistent with multiple alternative models. A separate line of research, using statistical methods to detect signals of admixture between genetically distinct populations in the ancient past, has found evidence in some northeastern North American populations of genetic signals that do not appear to be derived from the standard founding lineages of Native Americans. These signals are small. They are statistically significant, but not individually dramatic. And they are consistent in their geographic distribution with the hypothesis that they reflect very ancient Atlantic contact events, though they are also consistent with other explanations that do not require any cross-Atlantic migration. The probability that a single definitive paper will emerge and settle the question is low. Scientific consensus on questions of this kind builds slowly, through the accumulation of evidence from multiple independent lines of inquiry, each providing a small piece of the picture. The picture is currently incomplete, but it is becoming more complete every year, and the direction it is moving, toward a recognition of greater complexity and greater antiquity in the human genetic history of the Americas, is consistent with the mystery that the Rh-negative data in Canada represents. There is a type of evidence that sits outside the realm of DNA and archaeology entirely, and that is the evidence preserved in language. Languages carry history the way genes carry biology, in fragments and echoes that persist long after the people who created them have gone. And the languages of Indigenous Canada, when examined carefully and with an open mind, contain some very puzzling features that the standard models of North American prehistory do not easily account for. The Algonquian language family is the most widely distributed language family in North America, north of Mexico. It includes languages spoken from the Atlantic coast of Canada all the way to the Rocky Mountains, from the Gulf Coast of the United States to Hudson Bay and beyond. The Algonquian languages share a common ancestor, Proto-Algonquian, which linguists have reconstructed from their shared features, and they show the kind of systematic correspondence in vocabulary and grammar that indicates genuine common descent rather than borrowing. Proto-Algonquian was spoken, linguists estimate, somewhere between 2,000 and 4,000 years ago, meaning that the language family spread across most of temperate North America within a relatively short period of time. What is strange about Algonquian? What has puzzled linguists since it began to be systematically studied in the 19th century is the sheer geographic range of the family, combined with certain structural features that have no obvious parallels in other North American language families. The morphology of Algonquian languages, the way words are built up from smaller components to express complex meanings, is extraordinarily elaborate. Algonquian words can carry information that would require entire sentences in English, encoding not just grammatical relationships, but evidential information about how the speaker knows what they are claiming, spatial and temporal relationships between objects, and fine-grained distinctions about the animacy and intentionality of the things being described. Some linguists have argued that these features of Algonquian morphology represent a system so complex and so elegant that it could not have developed in a short time, that it reflects thousands of years of development in a relatively isolated linguistic community, and that the spread of Algonquian across such a wide geographic range was itself an unusual event that requires explanation. The usual explanation is population expansion, the spreading of a successful people across new territory along with their language. But the direction and starting point of that expansion have been debated. Some researchers have noted structural features in Algonquian that superficially resemble features in certain Celtic languages. The language family that includes Welsh, Irish Gaelic, Scottish Gaelic, Breton, and Cornish. These resemblances have been pointed to by proponents of pre-Columbian Atlantic contact as evidence of linguistic influence from Atlantic Europe. The mainstream linguistic establishment has been skeptical, because superficial structural resemblances between unrelated languages can arise through independent development rather than contact, and the specific features cited as evidence of Celtic-Algonquian connection are not detailed enough to constitute proof of relationship. But the question is not entirely closed. Linguists continue to debate the relationship between morphological complexity in language and the demographic and contact history of the populations that developed it. And within that debate, the specific case of Algonquian and its unusually complex morphological system remains an open question that not everyone is satisfied to explain, purely in terms of independent internal development. Let us also consider, for a moment, the vocabulary of the Algonquian languages as it relates to things maritime. The Algonquian languages have rich vocabularies for coastal environments, for maritime activities, for the specific features of the North Atlantic coast and its ecosystems. This is not surprising, since Algonquian peoples have inhabited the Atlantic coast for thousands of years and have developed their languages in close relationship with the sea. But some researchers have pointed to specific words and semantic categories in Atlantic Algonquian languages that appear to have no clear cognates in the more inland Algonquian languages, suggesting that they were either borrowed from another language or developed independently in the coastal communities. Whether any of those unusual coastal vocabulary items could represent borrowings from contact with Atlantic European populations, Norse or Basque or something older, is a question that has been asked but not answered. The methodology for detecting ancient loanwords in oral languages is difficult and contested, because the same phonological changes that transform languages over time also obscure the original forms of borrowed words, making them hard to distinguish from native developments. But the question itself is legitimate, and it sits alongside the blood type question and the haplogroup question as another thread pointing in the same direction. Among the most curious oral traditions in the Algonquian languages are the stories of the Wendigo. The Wendigo is a figure from Algonquian mythology who appears in stories across a very wide geographic range, from the Atlantic coast to the Great Plains. In different communities, the Wendigo takes different forms and plays different roles, but the common thread is a being of extraordinary size and power, associated with winter, with the north, with hunger, with the crossing of boundaries between the human and the inhuman. The Wendigo is in some versions a cannibal giant, in others a malevolent spirit, in others a kind of supernatural predator that preys on human souls as much as human bodies. The Wendigo stories are deeply embedded in Algonquian cosmology and have been interpreted in many ways, as cautionary tales about the dangers of starvation, as representations of the psychological effects of extreme isolation and cold, as mythological expressions of the unpredictability and danger of the northern environment. But some researchers have noted that the Wendigo figure bears certain resemblances to the giants and supernatural predators that appear in Norse mythology and in the mythology of other northern European peoples, specifically the frost giants of Norse tradition, beings associated with winter, with the north, with enormous size, and with a kind of primal wildness that threatens the ordered human world. This resemblance might be entirely coincidental. The northern environment, with its extreme cold, its long dark winters, its occasional famines, and its capacity to kill the unprepared, generates similar mythological responses in many cultures independently. The image of a monstrous cold being associated with winter is an obvious way for northern cultures to represent the threat of the environment. But the resemblance is noted, and it is one more thread in the web of suggestive connections between the indigenous cultures of northeastern North America and the cultures of the North Atlantic European world. There is also the matter of specific place names. The naming of natural features, rivers, lakes, mountains, and coastlines is one of the most persistent ways in which human history is preserved in landscape. When European colonizers arrived in North America, they found a landscape that already had names, and those names often survived in various forms in the European languages that eventually dominated the continent. But there are also places in Atlantic Canada where the name given by the original indigenous inhabitants, when translated, yields meanings that seem to describe features in ways that suggest knowledge coming from outside the immediate local environment, knowledge of distant places or traditions or peoples. Some researchers have pointed to place names along the Atlantic coast of Canada that appear to contain elements that could be interpreted as references to travelers from far away, to people who came from the direction of the sunrise over the ocean, to events involving the arrival of strangers. These interpretations are highly speculative and are not accepted by mainstream linguistics. Place names can carry many layers of meaning, and the temptation to read specific historical events into them is one that produces more fantasy than fact. But the persistence of certain themes in place names across the region, combined with everything else we have discussed, adds another faint line to the already complicated picture. Let us now return to the central question that started all of this. Why does Canada have Rh-negative blood frequencies that are higher than what the standard model of the Americas would predict? And after everything we have explored, what is the most honest answer we can give? The most honest answer is that there are multiple answers, at different levels of certainty, that together describe something more complex than any single explanation can capture. At the level of high certainty, Canada has elevated Rh-negative frequencies because a large proportion of its population is descended from Western European immigrants, who began arriving in the early 1700s and continued arriving in large numbers through the 19th and 20th centuries. Those Western European populations, particularly the French, the Scots, the Irish, and the Basque-influenced populations of southwestern France, carry Rh-negative blood at frequencies between 15 and 35%, which are among the highest in the world. When large numbers of these people settled a country and their descendants made up the majority of its population, the overall blood type frequency of the country reflected their genetic heritage. This is the bulk of the explanation, and it is not in dispute. At the level of moderate certainty, there may be a component of the elevated Rh-negative frequencies in certain Canadian communities that reflects pre-Columbian contact events, specifically the Basque whaling and fishing presence in Atlantic Canada in the late 1500s and possibly earlier, and the Norse presence in Newfoundland around 1,000 years ago. These contact events were too small and too brief to account for a large portion of the signal, but they may have introduced Rh-negative alleles into indigenous communities at a period before the main wave of colonial immigration, creating a small ancestral contribution that was then absorbed into both the indigenous gene pool and the colonial gene pool as they mixed over subsequent centuries. At the level of lower certainty but genuine scientific interest, there may be an even older component reflecting ancient Atlantic contact events in the deep prehistoric past, potentially going back to the maritime archaic culture, potentially going back to the Solutrean period, potentially involving people whose names we will never know and whose cultures have left no direct trace in the archaeological record except the faintest echo in the genetics of the people who descended from them. And finally, at the level of biological mechanism, the Rh-negative allele may have been maintained at somewhat higher frequencies in Canadian populations than simple demographic mixing would predict, due to some combination of founder effects in small isolated founding populations, genetic drift in communities that experience bottlenecks due to disease or displacement, and possibly a selective advantage that has not yet been fully characterized, but that may involve the particular immune characteristics that Rh-negative status confers. None of these answers alone is sufficient. None of them together constitutes a complete picture. The complete picture is still being assembled, one ancient DNA sample and one genetic database, and one carefully argued academic paper at a time. It will take decades. It will involve controversies and debates, and the slow, grinding consensus building that is how science actually works, as opposed to how it appears to work in popular accounts that want clean narratives and dramatic reveals. But science is not the only lens through which this question deserves to be examined. There is also the human lens, the personal and communal meaning that blood type has for the individuals who carry it and for the communities that have gathered around the shared experience of being. In a world of 85% Rh-positive majority, part of the 15% that is different. In Canada, As in many countries with diverse multicultural histories, questions of identity and belonging are particularly charged. Who is Canadian? What does it mean to belong to a place where the people who have been here longest are still recovering from the effects of colonial dispossession? What is the relationship between genetic heritage and cultural identity, and how should we think about the mixing and layering of different ancestries that makes up almost every Canadian family tree? Once you go back far enough, the Rh-negative question touches on these larger questions in ways that are not always comfortable. If pre-Columbian contact events introduced European genetic material into indigenous North American populations, does that change anything about the sovereignty or the cultural identity or the land rights of those populations? The answer is clearly no, and it is important to say so clearly. Cultural identity and sovereignty rights do not derive from genetic purity or genetic distinctness. They derive from the historical and contemporary reality of continuous cultural presence and political status. The genetic complexity of any human population, indigenous or not, does not undermine or enhance the rights of that population. Genetics is about ancestry. Rights are about justice. But the genetic complexity itself is worth understanding, not for political purposes, but because it tells a true story about what human beings do and have always done. We move, we meet, we mix. We carry each other's histories forward in our bodies without always knowing we are doing it. The people of Canada, all of them, carry in their blood the record of an extraordinary range of human movement and contact stretching back to the deepest human past. The Rh-negative marker is one small part of that record. It is interesting partly because it is one of the few genetic markers that is routinely measured and reported in a way that makes its distribution across populations visible to non-specialists. Most of the other genetic markers that tell the same kind of complex, layered story are buried in databases, accessible only to researchers who know how to look for them and how to interpret what they find. The fact that blood type is visible in this way, that people know their Rh status because it is clinically relevant and is noted in their medical records and on their blood donor cards, makes it a more accessible entry point into questions about human genetic history than most molecular markers. You can ask someone their blood type at a dinner party and get an answer. You cannot ask someone about their mitochondrial haplogroup and expect them to know it, or to be able to look it up easily. So, blood type, and particularly Rh status, has become a kind of shorthand for talking about genetic heritage, even though it is only one tiny piece of an incomprehensibly complex genetic picture. The mystery of Rh-negative blood in Canada is partly a mystery about blood, but it is mostly a mystery about time, and about how long human beings have been moving across this continent and across the oceans that border it, and about how much of that ancient movement we have lost track of in the relatively shallow records that written history provides. Written history in Canada goes back 400 years, to the first permanent European settlements. Indigenous oral traditions in Canada go back thousands of years, though interpreting what they say about the deep past is a process full of uncertainty. The genetic record in Canada goes back 10,000 or more years, though reading it is technically difficult and scientifically contested. And the full depth of human presence in Canada, the complete record of all the migrations and contacts and population events that produced the genetic diversity we observe today, goes back to the very beginnings of human presence in the Americas, to a time so distant that it is barely imaginable. 15,000 years ago, a small group of people stood at the edge of a glacier in what is now northern Canada and looked south into a landscape that had never heard a human voice. They had crossed from Siberia into Alaska, and they were the first human beings to stand on that particular patch of ground. Their blood was Rh positive, almost certainly, because they came from northeastern Asian populations where Rh negative blood was essentially absent. They had no idea what continent they were on. They did not know there was a continent there, in the sense we understand it. They were following animals or following coastlines or simply moving because human beings move, and ahead of them was an entire world with no people in it. Within a few thousand years, their descendants had reached the Atlantic coast of what is now Canada. Within a few more thousand years, they had spread across the continent from the Arctic to the tropics, adapting to every environment they encountered, developing technologies and languages and cultures of astonishing diversity. And at some point in that long history, something that the standard model does not fully account for happened. A bloodline carrying Rh negative alleles entered the gene pool of some of these populations, and that bloodline has persisted, diluted but detectable, down to the present day. Who those people were, when they arrived, where they came from, how they were received by the people already here, All of that is still, in the deepest sense, unknown. The mystery is real, the evidence is real, and the answer, when it finally comes in sufficient clarity to be recognized as an answer, will change the story of Canada and the story of the Americas in ways that we cannot yet fully anticipate. There are researchers working on this right now. In a laboratory at a Canadian university, a technician is carefully extracting ancient DNA from a piece of bone that spent thousands of years in the soil of a Nova Scotia midden. The sequencing machine is running. The data is accumulating. The statistician who will analyze it is at their desk, writing the code that will search for the pattern hidden in the noise. They may find something conclusive. They may find something tantalizing but ambiguous. They may find nothing that they do not already know. But they are looking. And the looking itself is a form of respect for the people whose blood has been flowing through this continent since before any record was kept, since before any map was drawn, since before anyone thought to ask where they came from or what they carried. The blood knows. It keeps the record, faithfully and without bias, indifferent to the stories we tell about it. All we have to do is learn to read it. And that, ultimately, is what makes this question about Canada and Rh-negative blood so compelling. It is not just a question about blood type frequencies or genetic markers or the specifics of who was where when. It is a question about what we do not know about ourselves, about our deepest history, about the extraordinary complexity of the human journey that produced every person alive today. Canada is a country that is still, in a very real sense, figuring out what it is and where it came from. The genetic mystery of its blood type distribution is one small piece of that larger project of self-understanding. The answer is not going to come all at once. It is not going to arrive as a single revelation that resolves the mystery cleanly and sends everyone home satisfied. It is going to come the way most important knowledge comes, slowly, in fragments, with persistent uncertainty at the edges, requiring constant revision and refinement as new evidence emerges and old interpretations are challenged. But it is coming. The science is getting there. And when it arrives, it will tell a story that is older and stranger and more human than anything we have yet imagined about the blood that flows through the bodies of the people who call Canada home. Consider what that means for the way we think about identity, about heritage, about the stories we tell ourselves, about where we come from. The standard Canadian narrative of a country built by two founding nations, French and English, on top of a land that was already inhabited by Indigenous peoples, is a narrative that has been challenged and complicated in countless ways over the past several decades. The recognition of Indigenous rights, the truth and reconciliation process, the growing understanding of the depth and sophistication of pre-contact Indigenous civilization, all of these have been working to complicate and enrich the simple founding narrative that Canada was told for most of its history as a modern state. The genetic evidence adds another layer to that complication. It suggests that the story of how Canada became populated, and by whom, and what connections those populations had to other parts of the world, is even more complex than the most nuanced political and historical accounts have recognized. It suggests that the genetic boundaries between populations that seem sharp and clear are in fact blurry, permeable, and ancient in their blurriness. It suggests that people have been moving back and forth across every border and every ocean for much longer than any written record acknowledges, and that the result of all that movement is a world in which nearly every person carries the genetic signatures of multiple ancestries reaching back to multiple continents. Canada's elevated Rh-negative frequencies are, from this perspective, not a mystery to be solved so much as a reminder to be absorbed. A reminder that the people on this land have been in contact with the rest of the world for far longer than anyone knew. A reminder that human history is more ancient and more interconnected than our maps and our records suggest. A reminder that the blood running through the veins of every Canadian, whether they are the descendants of Indigenous people who have been here for 15,000 years or the descendants of immigrants who arrived in the last generation, carries within it a record of extraordinary journeys and unexpected meetings, and the continuous, unstoppable human drive to move across the world and see what is on the other side. There is a specific community in Newfoundland that most people outside Canada have never heard of, and that represents one of the most concentrated and specific versions of the Rh-negative anomaly in the entire country. The community of Merasheen no longer exists as an inhabited settlement. It was resettled, which is the Newfoundland term for the forced relocation that the provincial government carried out in the 1960s and 70s, moving populations off small, remote outports and into larger towns in an effort to reduce the cost of providing services to geographically scattered communities. But before it was resettled, Maraschin was an isolated fishing community on Placentia Bay that had been continuously inhabited for several generations, with a tight-knit population descended from a small number of founding families. Communities like Maraschin, small and isolated over many generations, are where founder effects and genetic drift do their most dramatic work. When the founding population of a community is small, the frequency of any given genetic variant in that founding population becomes the frequency across the entire community, regardless of whether it is high or low compared to the broader population. If 2 or 3 of the founding families of Maraschin happen to carry Rh negative alleles at high frequency, then all of their descendants would carry it at high frequency. And since the community had limited contact with outsiders over many generations, there was little new genetic material coming in to dilute or alter that frequency. Studies of isolated Newfoundland communities have consistently found this kind of amplified genetic variation. The founder effect is so strong in some of these communities that they carry unusually high rates of rare genetic disorders that are almost unknown elsewhere, simply because the founding families happen to carry those rare alleles and pass them on to descendants who had limited opportunity to mate with people from outside the community. The same mechanism that amplifies rare disorders also amplifies whatever blood type frequencies happen to be present in the founders. This is a mundane explanation for the Rh negative anomaly in specific Newfoundland communities, and it is probably the correct explanation for those specific communities. But it does not explain why the founders of those communities had the Rh negative frequencies they did, which takes us back to the deeper questions about the source of the Rh negative alleles in the founding populations of Atlantic Canada. The founding populations of Newfoundland were diverse. They included West Country English fishermen who had been using Newfoundland waters since the early 1500s and who eventually established permanent settlements. They included Irish settlers who came in large numbers in the late 1800s and early 1900s. They included a scattering of French, Scots, and other European nationalities. And they included, in many cases, indigenous ancestry, primarily Mi'kmaq, but also Beothuk, in the communities of the northeast coast, which carries its own genetic complexity and its own uncertainties about deep pre-contact ancestry. The West Country English, from Devon and Cornwall and Somerset, are an interesting group. They are from the extreme southwest of England, a region that was relatively isolated from the Central English population for much of the medieval period, that had its own dialect and culture, and that genetically reflects the ancient Celtic population of Britain more than the later Anglo-Saxon or Norman influxes that shaped most of England's genetic profile. The people of Cornwall in particular show genetic affinities to the Welsh and the Breton populations of France, and carry haplogroups associated with the ancient Atlantic coastal population of Western Europe. Their Rh negative frequencies are somewhat elevated compared to the English average, reflecting that Celtic ancestry. So even in the West Country English contribution to Newfoundland's founding population, you have a genetic source that is biased toward the Western European, Celtic-influenced, Atlantic coastal end of the European genetic spectrum, which is exactly the end that carries the highest Rh negative frequencies. The founding of Newfoundland as a settled colony was not a random draw from the European gene pool. It drew disproportionately from the specific populations of Western Europe that fished the North Atlantic, and those populations were precisely the populations with the deepest genetic connections to the ancient Atlantic coastal tradition of Europe, the same tradition from which the Basque emerged, and the same tradition that the Solutrean hypothesis connects to the most ancient hypothetical crossings of the Atlantic. This is not a conspiracy. There is no hidden hand guiding genetics toward a predetermined conclusion. It is simply the result of the fact that the North Atlantic fishing industry was, for centuries, dominated by populations from the western Atlantic coast of Europe, and those populations were the ones who colonized the western Atlantic shores of North America. The genetic connection between the people on one side of the Atlantic and the people on the other side was, in part, a product of the fishing industry that connected them, which is not a romantic or mysterious explanation, but it is a human one. Let us think about the Grand Banks one more time, because it is central to this story in a way that is not always appreciated. The Grand Banks of Newfoundland are an underwater plateau, a section of the continental shelf where the cold Labrador Current meets the warm Gulf Stream, creating conditions that produce extraordinary biological productivity. The waters above the Grand Banks have historically been among the most fish-rich in the world, supporting populations of cod so large that early visitors to the region reported being able to lower a basket into the water and draw it up full of fish. This is probably an exaggeration, but the underlying reality is that the Grand Banks were an almost incomprehensibly rich resource, and that resource was the primary driver of European engagement with North America for a century before any permanent settlement was established. The people who exploited the Grand Banks were not primarily explorers or colonizers. They were fishermen, doing an industrial job in a dangerous environment, motivated by profit rather than adventure. They were the Basque, the Portuguese, the Bretons, the Norman French, the West Country English. They came every spring when the ice broke, they worked through the summer and fall, and they went home before winter. For much of the 16th century, the Grand Banks fishery was one of the largest industries in Europe, employing thousands of men and hundreds of ships, and the knowledge of the North American coast that those men accumulated over decades of annual voyages was practical, detailed, and transmitted through the informal channels of the fishing industry rather than through the formal channels of royal exploration. This means that the degree of contact between European fishing populations and the indigenous peoples of the Atlantic Canadian coast in the century before official colonial settlement was much greater than the historical record suggests. The historical record consists primarily of documents created by governments, churches, and literate elites, and the activities of working fishermen were simply not systematically documented. What was documented in the oral tradition of the fishing communities, in the practical knowledge of the captains and crews who returned year after year to the same bays and coves, has largely been lost. But the genetics are not lost, and the genetics of modern Atlantic Canadian communities, when analysed carefully, show the traces of those centuries of contact in the form of mixed ancestries and unusual genetic frequencies that do not fit any simple model of purely recent European immigration. One more thread deserves to be pulled before we begin to draw this long exploration toward its close. The question of what Indigenous Canadians themselves know, or have preserved in tradition about the ancient genetic history of their communities. This is a delicate subject. The imposition of Western genetic frameworks onto Indigenous identity has been used historically in ways that have caused real harm, including attempts to use genetic data to argue that Indigenous peoples were not sufficiently genetically distinct to constitute separate populations with land rights, or that European ancestry in Indigenous populations somehow diminished or complicated their Indigenous status. These uses of genetics are methodologically wrong, ethically abhorrent, and legally irrelevant to questions of Indigenous sovereignty. They deserve to be named and rejected clearly. But the question of what Indigenous oral traditions preserve about the ancient history of their communities, including possible ancient contact events with people from outside North America, is a different question, and it deserves to be engaged with respectfully and on the terms that Indigenous communities themselves set. Some First Nations scholars and community members have expressed interest in the genetic research questions surrounding pre-Columbian contact, not because they believe it would change their status or rights, but because they are interested in the full depth of their communities' histories. If some of their ancestors had contact with people from across the Atlantic, whether Norse sailors or Basque fishermen, or ancient people who crossed on ice 20,000 years ago, that is part of their history too, and knowing it more completely is a matter of historical truth rather than political convenience. Other community members and scholars are skeptical of the entire enterprise, concerned that the emphasis on pre-Columbian European contact is a way of implicitly questioning the distinctiveness or the indigeneity of their communities, or of distracting from the more pressing issues of land rights, cultural survival, and political recognition. These concerns are legitimate and they reflect a long history of exactly that kind of scientific bad faith. The challenge is to hold both of these things simultaneously, to pursue the genuine scientific questions about the deep genetic history of the Americas with rigor and without a predetermined agenda, while maintaining full respect for the sovereignty of Indigenous communities over their own biological and cultural heritage, and while refusing to allow the research to be weaponized in the political arguments that always surround questions of Indigenous rights and recognition. This is harder than it sounds, because science does not happen in a political vacuum, and the history of anthropology and genetics in relation to Indigenous peoples is a history of repeated violations of exactly this kind of boundary. But the alternative, which is to simply not ask the questions, to leave the genetic history of Canada permanently incomplete, rather than risk the political complications that come with asking, is also unsatisfactory. It is a disservice to the truth, and ultimately a disservice to the Indigenous communities whose histories are the most important part of the story. Let us spend some time with the specific mechanics of how a genetic variant, like Rh negative, would actually move through a population, because understanding the mechanics helps clarify both the possibilities and the limits of what we can infer from the data. When a new genetic variant appears in a population, either through mutation or through introduction by migrants from another population, its subsequent frequency depends on two factors: natural selection and genetic drift. Natural selection favors variants that increase reproductive success and disfavors variants that decrease it. Genetic drift is the random fluctuation in allele frequency that occurs in all finite populations but is especially strong in small ones, where chance events in reproduction can shift allele frequencies dramatically from one generation to the next. For the Rh negative variant, the selective landscape is complicated. As we discussed earlier, Rh negative is associated with a reproductive disadvantage in heterozygous pregnancies because an Rh negative mother carrying an Rh positive fetus can mount an immune response that endangers subsequent fetuses. This is a real selective cost. In the absence of medical intervention, it would be expected to exert downward pressure on Rh negative frequencies in any mixed population over time. But Rh negative frequencies in Western Europe have not been declining. They have been roughly stable for as long as we can measure them, which in the case of populations with good historical records means several centuries. This stability, despite the reproductive cost, implies either that there is a countervailing selective advantage, or that the reproductive cost has been smaller in practice than the theory would predict, or that some combination of factors has been maintaining the frequency at a rough equilibrium. The selective advantage hypothesis is the most interesting scientifically. What advantage might Rh negative blood confer that would offset the pregnancy cost? The most extensively studied candidate is resistance to certain parasites. The Rh antigen, the protein whose absence defines Rh negative blood, appears to be involved in certain aspects of how red blood cells interact with their environment, and some parasites exploit red blood cell surface proteins as binding sites for infection. An Rh negative individual, lacking the Rh antigen, might present a modified cell surface that certain parasites cannot bind to as effectively, conferring some degree of resistance. Research on this question has been conducted in the context of malaria, which exploits red blood cell surface proteins extensively in its life cycle. Some studies have found differences in malaria susceptibility between Rh positive and Rh negative individuals, though the results are mixed and the effect sizes are small. Research in other parasite contexts, including toxoplasmosis and certain helminth infections, has also found some Rh-related differences in infection rates or outcomes, again with small effect sizes and inconsistent results across studies. The smallness of the measured effects does not rule out a meaningful selective advantage. Even a very small selective advantage, compounded across many generations, can explain the maintenance of a genetic variant at a given frequency. A 1% per generation advantage in survival or reproduction is invisible on any human timescale, but it is more than sufficient to explain why a variant that causes a measurable reproductive disadvantage in one context is nevertheless maintained at stable frequencies across hundreds of generations. For Canada specifically, the question of whether Rh-negative individuals showed differential survival during the epidemic periods of the colonial era has never been properly studied, partly because the data does not exist in the right form and partly because it has not been a priority. The epidemic mortality of indigenous populations in particular was so catastrophic and affected so many aspects of those communities' demographic profiles that identifying a blood type specific component of mortality within that catastrophe would require an extraordinary quality of historical and genetic data that simply does not exist for the relevant time periods. But here is an interesting thought experiment. If Rh-negative individuals, whether indigenous or European origin, showed even a modest advantage in surviving the major epidemic diseases that swept through North America in the 17th and 18th centuries, the post-epidemic populations would have higher Rh-negative frequencies than the pre-epidemic populations. This is the standard mechanism by which epidemic disease creates genetic selection. The survivors pass on their genes, and if those genes include frequencies of a variant that conferred some survival advantage, the next generation carries that variant at a higher frequency than the previous generation. Over several generations of such selection, the Rh-negative frequency could have been pushed significantly higher in populations that survived major epidemic events. If those populations included both European origin settlers and indigenous communities with some existing Rh-negative frequency from ancient contact events, The post-epidemic populations in both groups would have elevated Rh negative frequencies that do not simply reflect the pre-epidemic demographic input. This mechanism does not require ancient pre-Columbian contact to be real. It operates within the documented historical period. But it does suggest that the Rh negative frequencies we measure in contemporary Canadian populations may not simply reflect the weighted average of the source populations that contributed to that contemporary population. They may also reflect the selection events that shaped who survived and who reproduced during the critical periods of colonial-era demographic catastrophe. This is, again, a deeply uncomfortable hypothesis if you follow its implications to their logical conclusion. It suggests that some of the highest Rh negative frequencies in Canada might be concentrated in communities with the most complex histories of epidemic exposure, which means communities in the regions that had the earliest and most intensive contact with European populations and their associated diseases. It suggests a biological footprint of colonialism that runs deeper than cultural dispossession and material deprivation, all the way into the genetic profile of surviving populations. This is not a unique insight in the context of population genetics. Researchers studying other selective forces, including famine, warfare, and climate, have found genetic signatures of historical catastrophes in many human populations. The idea that the demographic catastrophes of the colonial era left genetic signatures in North American populations is not novel, and the specific hypothesis that blood type played a role is speculative but not implausible. What would be needed to test this hypothesis properly is a comparison of Rh negative frequencies in pre-contact ancient DNA samples from relevant North American populations with Rh negative frequencies in post-contact populations from the same regions, combined with epidemiological data on the specific epidemic events those populations experienced. This is precisely the kind of research that the growing availability of ancient DNA is beginning to make possible. And it is the kind of research that, when it is eventually conducted at sufficient scale and with sufficient resolution, will produce answers to questions that are currently open. In the meantime, the mystery persists. The blood type databases show what they show. The genetic studies produce the results they produce. And the gap between what the data shows and what any single clean explanation can account for remains, pointing towards something in Canada's deep history that we have not yet fully understood. There is a particular kind of intellectual pleasure in sitting with a genuine mystery, not the fake mysteries manufactured for entertainment, where the mystery is designed to be solved in a satisfying reveal at the end, but real mysteries, the ones where the evidence is real and the questions are legitimate and the answers are not yet known. These are the mysteries that drive science forward, that keep researchers coming back to the same datasets with new methods, that generate the papers and the conferences and the quiet debates in university hallways that eventually produce the slow, grinding progress of knowledge. The mystery of Rh negative blood in Canada is that kind of mystery. It is not a mystery that can be solved in an afternoon, or a documentary, or a YouTube video. It is a mystery that will be solved, partially and incrementally, over decades of careful research by dozens of scientists working on different pieces of the same puzzle, occasionally recognizing that their piece fits with someone else's piece in a way that neither of them had expected. And it is a mystery that matters. Not primarily because it might reveal ancient lost civilizations or secret European knowledge of North America that rewrites history in a dramatic way, though those possibilities, however remote, cannot be entirely ruled out. It matters because it is one more reminder of how much we do not know about the most fundamental questions of human history. Who are we? Where did we come from? How did we get here? What connections did we have to people in distant places before any written record began to track our movements? These are questions that every human civilization has asked, and that no civilization has ever fully answered. The genetic data is giving us new tools to approach those questions with greater precision than was previously possible. But precision is not the same as completeness. The human past is vast, and the genetic record, extraordinary as it is, captures only a fraction of what happened. The rest is silence. The rest is the absence of evidence that cannot be distinguished from evidence of absence. The rest is the ocean floor, 300 feet of cold Atlantic water sitting on top of the villages where people lived and loved and died and passed their blood forward, not knowing that their descendants would one day be puzzled by the traces left behind. Something happened in Canada's deep past that we cannot fully account for. That is the plain statement of the case. Everything else in this exploration has been an attempt to characterize the shape of that something. to trace its outlines from the inside, using the limited tools available to us, to understand not just what is known, but what remains unknown and why it remains unknown, and what it would take to know it better. The shape that emerges from everything we have discussed is not one thing, it is multiple things, layered and overlapping, each contributing something to the picture. There is the deep prehistoric peopling of the Americas, with its standard story of Beringia crossings and its increasingly complicated reality of multiple migration events, possible Atlantic contact, and genetic diversity in the founding populations that is no longer fully preserved in modern indigenous Americans. There is the maritime archaic culture, those ancient seafaring people of the Atlantic Canadian coast who used red ochre in their burials, hunted sea mammals in the open ocean, maintained long-distance trade networks across an enormous geographic range, and whose genetic profile remains largely unknown because the acidic soils of the Atlantic coast have consumed their bones. There is the old copper complex, those ancient miners of the Great Lakes who extracted hundreds of millions of pounds of copper from the shores of Lake Superior and distributed it across the continent through trade networks that must have involved sustained movement of people as well as goods. There is the Norse settlement at Lanceau Meadows, confirmed by archaeology and by the sagas, representing a documented contact event between northern European people and the indigenous inhabitants of Newfoundland, with its potential for genetic exchange that the available data has not yet been able to confirm or deny. There is the Basque presence in the waters off Atlantic Canada, earlier and more extensive than the historical record fully captures, represented archaeologically at Red Bay and documented genetically in the burial remains of Basque sailors who died on the Labrador coast. There is the founding of French and British Canada by populations drawn disproportionately from the Western European populations, with the highest Rh negative frequencies, creating a strong recent signal that dominates the modern Canadian genetic profile, but cannot by itself explain all the features of the data. And running through all of these, connecting them and complicating them, and refusing to resolve into any clean narrative, is the blood. The Rh negative blood of the people who have lived on this land for every period of this story, indigenous and European and mixed, carrying in their cells the record of every contact event and every migration and every generation of survival and reproduction since the first human being set foot on the cold shores of what would one day be called Canada. What does it all mean? For most people, it means very little in practical terms. Knowing that Canada has somewhat elevated Rh negative frequencies does not change anyone's daily life or alter any political or legal reality. The blood type data is a curiosity, an interesting number in a medical database, a starting point for questions that most people never actually ask. But for the people who ask those questions, the mystery of Canada's blood is an entry point into the full depth and complexity of the human story on this continent. It is a reminder that the history of North America, like the history of every place human beings have inhabited, is older and stranger and more complicated than any simple narrative can capture. It is a reminder that we are all, in every place and every culture, the product of movements and meetings and mixings that go back far further than any written record, far further than any oral tradition, to the very origins of our species, and its extraordinary drive to move across the surface of the planet and carry its genes, its languages, and its blood into every corner of the world. Canada is a country that is still figuring out its own story. The debates about its history, about who was here, what was done to them, and what obligations that creates for the present, are live and ongoing and deeply important. The genetic mystery of its blood type distribution is one small piece of those larger debates, and it does not resolve them or simplify them. If anything, it makes them more complex, by suggesting that the human history of this country is even longer and more entangled than the most complicated political account has yet acknowledged. But complexity is not a problem. Complexity is the truth of human history, and coming to terms with that complexity, being willing to sit with it rather than resolve it prematurely into a simple story, is a mark of intellectual maturity and historical honesty. The simple stories about where we come from and who we are have always been inadequate to the actual richness of the human past. The genetic data is one more tool for peeling back those simple stories and seeing the complexity underneath. There is beauty in that complexity. There is something almost vertiginous about looking back through 15,000 years of human presence in Canada and seeing the extraordinary range of people and cultures and contacts that produce the country that exists today. The Rh negative question is, ultimately, a question about connection, about the connections between people across oceans and centuries and millennia. about the way human beings have always found each other, across every distance and every barrier, and left traces of those meetings in the most durable record available, the living DNA of their descendants. Those descendants are alive today in every province and territory of Canada, carrying in their blood the accumulated evidence of everything that happened here before history began to be written. They are the archive, they are the story, and the story is not finished. It is still being written, in the lab and in the field and in the careful, patient work of researchers who are asking, one question at a time, where Canada's blood came from. The answers, when they come, will add new chapters to a story that is already extraordinary, and that will become more extraordinary still as we learn to read it more completely. The mystery is real, the blood is real, and somewhere in the space between what we know and what we do not yet know, in the gap between the data we have and the data we need, in the silence of the drowned villages and the unrecorded encounters and the ancient migrations that left only the faintest trace, the answer is waiting. It will be found, not all at once, not by a single brilliant discovery, but piece by piece, in the slow accumulation of knowledge that is how human beings have always come to understand their own history. And when it is found, it will be not the story of any one people or any one contact event or any one blood type marker. It will be the story of all of us, reaching back across the full depth of human time on this extraordinary continent, and finding in that depth not separation and distinctiveness, but connection. The ancient, persistent biological fact of human connection, recorded in the blood, preserved in the genes, and waiting for us to read it clearly enough to understand what it means. Canada has more Rh-negative blood than almost any country in the Americas. The origin is unknown. And that unknown, properly understood, is not a gap in our knowledge but an invitation to look deeper, to ask harder questions, to resist the simple answers that do not fit the evidence, and to follow the blood wherever it leads. It leads further than we know. It leads further than we have yet looked. It leads back to the very beginning of the human story on this continent, and possibly to connections across the Atlantic that predate any history we have written, to people who crossed ice or ocean when the world was very different, and who left behind, in the blood of those who came after them, the only record we have of the fact that they were there. That record is still speaking. We are only beginning to learn how to listen, and there is something in that, something in the act of listening carefully to what the blood is trying to tell us, that feels like the most important kind of historical work. Not the construction of grand narratives or the defense of established interpretations, but the patient, rigorous, humble attention to evidence that does not fit any existing story, and the willingness to let that evidence change the story rather than forcing it to conform to what we already believe. Canada's Rh-negative blood is that kind of evidence. Sitting quietly in the medical records and the genetic databases, not demanding attention, not announcing itself as important, simply being what it is. A number that is slightly larger than the models predict. An anomaly that has been waiting, patiently, for someone to ask why. The question has been asked. The answer is still coming. And the story that answer will eventually tell is going to be one of the most interesting stories in the long, strange, extraordinary history of human beings on this planet. That much, at least, we can say with confidence. Let us take one more walk through the specific geography of the question, because geography is ultimately what ties all of these separate threads together, and because the map of Canada's Rh-negative distribution, when you look at it carefully, tells a story that no single explanation captures, but that every explanation we have discussed contributes to. The Atlantic coast is the beginning. Nova Scotia, New Brunswick, Prince Edward Island, Newfoundland, and Labrador. These are the places where the Old World and the New World have been closest together for the longest time. These are the places where the fishermen came first, and where the settlers followed, and where the mixing of people from the east with people from the west has been going on for longer than any documented record confirms. The Rh-negative frequencies here are high, and they are high for reasons that include everything from Basque whaling culture to Scottish Highland genetics to the deep pre-contact history of the Micmac. They are the highest layer of a palimpsest that contains many older layers beneath. Moving inland, up the St. Lawrence Valley and into Quebec, the frequencies remain elevated, carried by the founding French-Canadian population and maintained by the genetic isolation and drift that affected many Quebec communities over the centuries of agricultural settlement. Quebec carries its Rh-negative frequencies like a genetic signature of its origins in the fishing villages of northwestern France and the Basque-influenced communities of the southwest, and in the complex early colonial mixing with the indigenous peoples of the Strait, Lawrence Valley. Moving further west, into Ontario and the Great Lakes region, the frequencies begin to reflect the more diverse origins of the populations that settled there. English, Scottish, and Irish settlers, plus later waves of immigrants from Central and Eastern Europe, produce a distribution that is somewhat lower than the Atlantic coastal average but still well above the East Asian baseline. And underneath the settler genetic profile, in communities with indigenous ancestry, there are the traces of the ancient people of the Great Lakes, the people who built the copper networks and the woodland mounds and the agricultural villages that European settlers found and destroyed and replaced. Into the prairies and the mountain west, the demographic history changes again. Ukrainian and Scandinavian and German farmers brought different genetic profiles than the Atlantic coastal settlers, and the indigenous peoples of the Western Plains had different histories and different contact patterns than the Eastern peoples. The Rh-negative frequencies reflect these differences, being somewhat lower than in the East, but still higher than the global baseline that an East Asian founding population alone would produce. In British Columbia, the meeting of Pacific coastal indigenous cultures with settlers from across Europe and Asia produces yet another genetic mixture, one that is more influenced by the Pacific Rim populations who have been a significant part of British Columbia's demographic history since the 19th century, and that shows Rh-negative frequencies that reflect that more diverse input. And in the north, in the territories, the frequencies drop toward the baseline levels characteristic of the indigenous Arctic and subarctic populations, who descended most directly from the ancient migrations from Siberia, and who have had the least admixture with the Rh-negative carrying European populations. This geographic gradient, from high in the east and Atlantic coast to lower in the west and north, is not a surprise. It exactly mirrors the pattern of European settlement and indigenous European mixing, with the areas of most intensive and earliest European contact showing the highest frequencies. But it is the specific level of the frequencies in specific communities, and the specific pattern of which communities deviate from what simple demographic mixing would predict, that keeps the questions open. Because the gradient is not smooth, there are pockets of unusual concentration that stand out even against the general elevated background of Atlantic Canadian genetics. There are specific Mi'kmaq communities, and specific Acadian communities, and specific communities with deep Scottish or Basque roots, where the Rh-negative frequencies are higher than any simple model of recent European immigration would predict, even accounting for founder effects and drift. And those specific pockets are the places where the deeper questions become most pressing. If you wanted to design a research program that had the best chance of resolving the mystery of Canada's Rh-negative anomaly, you would start with those specific communities. You would conduct ancient DNA analysis on the earliest preserved human remains from the region, trying to extend the genetic record back into the pre-contact era. You would do detailed population genetic analysis of modern community members, with the full consent and participation of the communities themselves to characterize the full complexity of their ancestry. You would combine that genetic analysis with detailed historical and archaeological research to understand the specific contact events and migration patterns that shaped each community. And you would bring it all together in a statistical framework that could distinguish between the different possible explanations for the elevated frequencies you observe. That research program does not yet exist in its full form. Pieces of it are being conducted at various institutions, by various researchers, with various funding sources. The pieces are not always well coordinated. The political and ethical constraints on working with indigenous genetic data mean that some of the most important pieces are not yet available. And the scientific community has not yet reached a consensus on how much weight to give to the anomalous signals in the data versus the simpler explanations that account for most of the variation. But the question is being asked, in more sophisticated ways and with better tools than ever before. And the answer, when it comes, will be worth waiting for. While we wait for that answer, there is another way of engaging with this mystery that is not scientific, but that is no less real. It is the way of the imagination, the willingness to inhabit the specific moments and specific people that the historical and genetic evidence points toward, to try to understand not just the statistical pattern, but the human reality that the statistics represent. Imagine a Basque sailor in the 1530s, anchoring in a cove on the southern shore of Labrador for the winter, too late in the season to make the crossing back to Europe, taking his chances with the cold and the isolation, spending months in close contact with a Mi'kmaq family who found him and chose to keep him alive, rather than let him die in the snow. Imagine the conversation that must have happened, in sign language and borrowed words, and the universal language of shared work and shared meals, as the Basque sailor and the Mi'kmaq family navigated the extraordinary strangeness of their encounter. Imagine the child that may or may not have been born from that encounter, and what that child's life was like, and whose descendants are alive today without knowing anything about a Basque sailor who spent a winter in Labrador 500 years ago. Imagine the Norse woman Gudrid, who the saga of Eric the Red says gave birth to a son named Snorri in North America around 1,000 years ago, the first European child born in the New World. What happened to Snorri? The sagas say he returned to Europe with his mother and lived a long life there. But what if some of the Norse stayed, or came back, or left more children behind than the sagas recorded? The sagas were not comprehensive. They were stories, shaped by memory and storytelling conventions, and they recorded what their tellers thought was worth recording. The genetic traces of people who chose to stay would not have been recorded in any saga. They would only have survived in the blood. Imagine someone who crossed from Europe to Canada long before the Norse, following the edge of the ice in a world that looked nothing like any world we recognize, hunting seal on the frozen ocean, navigating by stars that have since moved from the positions they held then, carrying their blood westward into a land that no European, in any history we know, had ever reached. We cannot name this person. We cannot describe their face or their culture or their language, but they may have existed, and if they did, their descendants are here, in Canada, carrying in their blood a trace of a journey so ancient that no human memory has preserved it. This is what makes the mystery of Canada's blood so haunting and so beautiful. It is not just a question about genetics, or history, or archaeology. It is a question about the depth of human time on this continent, and about the extraordinary complexity of the human story, and about the way that the past persists in us long after we have forgotten it. Every person alive is a living archive of human history, stretching back hundreds of thousands of years, and most of what that archive contains has never been read or recorded or understood. The Rh-negative question in Canada is one small attempt to read that archive in one specific place, for one specific marker, at one specific moment in the development of the science of human genetics. It is a beginning, not an ending. It opens more questions than it answers, and it points toward a future in which we will understand much more about where we come from than we currently do, not because any single discovery will reveal everything, but because the slow accumulation of knowledge that is how science works will eventually build a picture that is more complete, more accurate, and more extraordinary than anything we can currently imagine. The blood is the beginning. The mystery is real, and the journey toward understanding, like all the journeys that brought human beings to this continent in the first place, is one worth taking. Now let us revisit something that has been sitting at the edge of this discussion throughout, something that most researchers are careful to acknowledge, but that popular accounts of the Rh-negative mystery often either exaggerate beyond recognition or dismiss entirely without adequate engagement. This is the question of what the anomalous Rh-negative frequencies in Canada actually feel like to the people who carry them. This is not a scientific question, it is a human question, and it deserves a human answer. There are people in Canada, thousands of them, who have discovered that they are Rh-negative and have then gone looking for what that means, and who have found, in the process of that looking, a community of people who share a particular kind of curiosity about their own origins. These are not people who believe in ancient astronauts or in blood-type superpowers. Many of them are exactly the kind of thoughtful, skeptical, curious people who would recognize the difference between a serious scientific question and a fringe theory. They are people who simply want to understand something unusual about themselves, and who have found that the science is incomplete, and the history is contested, and the mystery is real. For some of these people, the Rh-negative question becomes a lens through which they explore their own genealogy, tracing their family trees back through the generations of settlers and immigrants, and mixing that produced them. They find French-Canadian grandparents and Scottish great-grandparents and Irish great-great-grandparents, and they trace those lineages back to the specific regions of France and Scotland and Ireland where Rh-negative frequencies are highest. They find, in some cases, indigenous ancestors who appear in the family tree in the ambiguous ways that mixed ancestry often appears in the records of a colonial society. And they find, at the end of their genealogical reach, the blank wall of the unrecorded past, the place where the family tree runs out of documented branches and merges with the undifferentiated genetic heritage of all the people who came before. The Rh negative marker for these people is a thread that might lead further than the documents can take them. It might connect them, through the genetic record, to people and places and histories that no document has recorded. The science does not yet have good enough tools to follow that thread all the way back, but the desire to follow it is not irrational or mystical. It is the ordinary human desire to know where you come from, applied to a time before written records began. In Quebec specifically, where the genetic genealogy of the founding French-Canadian population has been studied in extraordinary detail, this desire has produced a remarkable body of community-engaged research. The Quebec Family History Society and similar organizations have built databases connecting hundreds of thousands of modern Quebecers to their founding ancestors in the early colonial period, creating one of the most detailed genetic genealogical records in the world. The researchers working on these databases have been able to trace specific genetic variants, including blood type alleles, back to specific founding families and specific regions of France. And in some cases, they have found that the genetic profiles of founding families do not match any known French regional population, as well as they match the profiles of the Basque and Basque-influenced communities of southwestern France. This is interesting for a straightforward demographic reason. The historical record shows significant Basque participation in the early French colonization of Canada, particularly in the maritime and trade sectors. But it becomes more interesting when you look at the frequencies and find that even accounting for the known Basque contribution to the French-Canadian founding population, there are some genetic features, including blood type frequencies, that the known historical migration cannot fully explain. The gap is small. It is not dramatic. It is the kind of gap that could be explained by sampling error, or by the imprecision of the demographic data, or by the founder effects that amplify any frequency in a small founding population. But it is there, and it is consistent across multiple independent analyses, and it adds one more small weight to the pile of evidence that the full story of how Rh-negative blood reached its current frequencies in Canada involves something older than 400 years of European colonization. There are researchers in Canada who are specifically focused on this question right now, combining the detailed genealogical databases of Quebec and Atlantic Canada with modern and ancient genetic data to try to trace the Rh-negative signal as far back as the evidence will allow. Their work is slow, careful, and conducted with the kind of methodological rigor that is appropriate for a question with this many possible confounds and this many politically sensitive dimensions. They are not announcing dramatic discoveries. They are building a picture, incrementally, one study at a time. The picture they are building is not yet complete. It may not be complete for another decade or two, but the direction it is pointing is toward a story of Canada's genetic history that is richer and deeper and more complex than anything that has been publicly acknowledged in any mainstream account of the country's past. The Rh-negative mystery is, in this sense, a small window into a much larger project. The project of understanding what actually happened in Canada before the Europeans arrived with their ships and their diseases and their written records, and what the relationship was between those pre-contact events and the Canada that exists today. That project is ongoing. It is being conducted in laboratories and archives and archaeological sites and community meetings across the country, and it is producing, slowly, a picture of Canada's deep history that is going to be extraordinary when it is finally complete. For now, the mystery remains. The blood type frequencies are real. The gap between what the data shows and what the standard explanations predict is real. And the questions that gap opens are real questions, worth asking seriously, worth pursuing patiently, and worth sitting with even in the absence of clean answers. That is the nature of real mysteries. They do not resolve quickly. They require patience and rigor, and the willingness to live with uncertainty for as long as it takes to accumulate enough evidence to see clearly. Canada's Rh-negative blood has been waiting for this kind of attention for decades, and it is only now beginning to receive it in the form of the sophisticated genetic and archaeological research that the question demands. The answer is coming. It will take time. And when it arrives, it will be one of the most interesting answers in the history of a country that is still learning to understand its own deepest history. In the meantime, the blood runs on, through the arteries and veins of everyone alive in Canada today, carrying the record of every journey and every meeting and every generation of survival that produced them. Rh-negative or Rh-positive indigenous or European or Asian or African, or mixed in ways that no single category captures. Every person in Canada is a chapter in a story that goes back further than any written record to the very first time a human being stood on this continent and began the long process of making it home. That story belongs to everyone. It is the most inclusive kind of history, because it is written in biology rather than politics, and because biology does not recognize the distinctions between groups that politics requires. The blood does not care who owns the land or who has the right to be here. It only carries the record of who was here and for how long, and who they met when they arrived. Canada has more Rh-negative blood than almost any country in the Americas. The origin of that excess remains unknown, at least in its deepest form, and that unknown is not a failure of knowledge. It is an invitation to look deeper, to ask harder questions, to pursue the truth of this country's extraordinary genetic history with the same rigor and curiosity and humility that the best science always brings to the hardest problems. The invitation is open, the mystery is real, and somewhere in the vast, cold, ancient, beautiful country that is Canada, the answer is waiting. But we are not done yet. Because there is one more thread in this web of mystery and evidence and speculation that has not yet been fully pulled, and it is the thread that connects the Canadian Rh-negative question to one of the most fundamental debates in the history of American archaeology, a debate that has been running for more than 30 years and that shows no sign of reaching consensus anytime soon. This is the debate about the first Americans, about when exactly the first human beings set foot on this continent, how they got here, and what they found when they arrived. The conventional timeline, which held almost unchallenged through most of the 20th century, places the first Americans arriving from Siberia via the Beringia land bridge between 13,000 and 15,000 years ago, at the end of the last glacial maximum, when the ice sheets had retreated far enough to open a path south through the interior of Canada. This model is called the Clovis First model, after the distinctive stone tool tradition found at sites across North America that radiocarbon dates to between 12,500 and 13,000 years ago. For decades, no one could find convincing evidence of human presence in the Americas before the Clovis culture, and the Clovis First model held. Then the evidence began to crack. Monteverde in Chile, 1,500 kilometers south of the nearest Clovis site, produced radiocarbon dates of 14,500 years ago, which means the people who made those tools were in South America before the ice-free corridor through Canada was even open. This is not possible if the Clovis First model is correct, and so the model had to change. The most widely accepted modification is the coastal migration theory, which holds that the first Americans came not through the interior of Canada, but along the Pacific coast, island hopping and kelp forest harvesting their way from the Bering Sea to the tip of South America faster than any overland route would have allowed. The coastal migration theory is compelling, and it has significant genetic support in the form of the Pacific coast, indigenous genetic profiles that look most like the founding population of the Americas. But it still assumes a Northeast Asian origin for all American ancestry. It revises the root, not the source. And the root revision, while important, does not address the anomalous features of Eastern North American genetics that have been the focus of this discussion. Because if the coastal migration along the Pacific came from the same Northeast Asian source population as the interior Beringia crossing, neither of them accounts for the West Eurasian genetic signal that appears in the oldest Eastern North American remains, the haplogroup X that concentrates in the northeastern United States and Canada, or the blood type anomalies that we have been tracing throughout this exploration. Those anomalies require a different source, and the coastal Pacific migration route does not provide it. The population that could have provided it was somewhere in the North Atlantic, and the most plausible candidates are the Ice Age populations of northwestern Europe, specifically the Solutrean and related cultures that occupied the Atlantic coast of Europe from the Iberian Peninsula to the British Isles during the last glacial maximum. These populations were not primitive. They were sophisticated hunters and tool makers with a deep knowledge of the coastal and maritime environment. They were the ancestors of the Basque and of the ancient Celtic populations of the British Isles, and they carried haplogroup X and Rh-negative blood at frequencies far higher than any East Asian population. The idea that some of these people crossed the North Atlantic via the ice edge is called the Kelp Highway hypothesis in its Pacific version, and the Ice Highway hypothesis in its Atlantic version. The Atlantic version is far more controversial than the Pacific version, partly because the distances involved are larger and the evidence is thinner, and partly because it has been associated with some researchers and some popular accounts that have used it to make claims about European priority in the Americas that carry uncomfortable political overtones. But the hypothesis can be evaluated on its scientific merits independently of its political associations, and when it is, it turns out to be more plausible than mainstream archaeology has generally acknowledged. The distances involved in the North Atlantic crossing, while large, are not impossibly large for people with the maritime technology and environmental knowledge that the Ice Age populations of Western Europe possessed. The sea ice that covered the North Atlantic during the last glacial maximum would have provided a surface on which people could travel, hunt, and camp, using skills that Arctic hunters have possessed for thousands of years. The resources available along the ice edge, primarily seals, walruses, and seabirds, would have been sufficient to sustain a travelling population. And the genetic and archaeological anomalies in northeastern North America are consistent with, though not proven by, a founding contribution from such a crossing. The critical question is not whether such a crossing was physically possible, it clearly was, but whether there is sufficient direct evidence to conclude that it actually happened. And here the honest answer is that the evidence is suggestive but not definitive. The haplogroup X distribution, the blood type anomalies, the morphological diversity in the oldest North American skeletal remains, the Solutrean-looking stone tool assemblages on the Chesapeake coast, all of these point in the same direction. But none of them individually constitutes proof, and the combination of them together is consistent with the hypothesis without being required by it. This is the state of the question. Not proven, not disproven, sitting in the scientifically legitimate but uncomfortable space of unresolved controversy. The researchers who take it seriously are not fringe theorists. They are working scientists at established institutions, publishing in peer-reviewed journals, conducting the kind of careful research that is the appropriate response to a genuine scientific puzzle. The researchers who are skeptical have legitimate reasons for their skepticism and are making reasonable methodological demands for more and better evidence. Neither side is obviously wrong, and the debate is the kind of productive scientific controversy that is how knowledge advances. For Canada, the relevance is direct. If any Atlantic crossing occurred in the deep past, the eastern shores of North America were the landfall. The maritime archaic people, whoever they were and wherever they came from, were living exactly where such landfall would have occurred. The genetic anomalies in the people of Atlantic Canada are exactly the kind of signal that such a crossing would have left. And the geographic distribution of the anomalies, concentrated in the northeast and attenuating as you move west, is exactly what you would expect from a founding event on the eastern coast that then spread inland through the trade and migration networks of the continent. This does not prove that Atlantic crossing happened, but it provides a geographic and demographic context in which such a crossing would fit the evidence we observe, and in which its absence leaves the evidence unexplained. The genetic research of the next decade will be the best opportunity we are ever likely to have to resolve this question. The ancient DNA databases are growing faster than anyone would have predicted 10 years ago. The methods for analyzing small genetic signals in ancient samples are improving at an extraordinary rate. The indigenous communities in Atlantic Canada are engaging with genetic research in increasing numbers on their own terms and for their own purposes, creating the possibility of community-authorized studies that could access the most important samples. If the answer is out there, hidden in the DNA of the people who have been on this land since before any memory or record began, the next generation of researchers will find it. They will find it not in a single dramatic discovery, but in the slow accumulation of evidence that eventually tips the scale from unresolved to understood. And when that tipping point comes, the story of Canada and the story of the Americas will be changed. Whether that change will be dramatic or subtle, whether it will rewrite the textbooks or merely add a footnote, is impossible to predict from where we stand. The history of science is full of cases where the answer to a long-standing mystery turned out to be more complicated and more interesting than any of the hypotheses that preceded it. And this question has the feel of one of those cases. The mystery is real enough that the answer, when it comes, is unlikely to be simple. Until then, the blood carries the record forward, faithfully and without judgment, through the arteries and veins of every person alive in Canada today. Rh negative and Rh positive, ancient ancestry and recent immigrant, indigenous and settler, and everything in between that defies those categories. All of them carrying, in the cells of their blood, the accumulated evidence of every journey and every meeting and every generation of survival that produced the extraordinary, complex, still being discovered country that Canada is, that is the mystery. That is the invitation. And that is why the question, Canada has more Rh negative blood than almost any country in the Americas, is worth asking in full, and worth following wherever the evidence leads. And so we arrive at the place where all long journeys eventually arrive, the place where you have to decide what to do with everything you have learned along the way. Canada is easy to reduce to a set of symbols, maple leaves and hockey and mountains and politeness. But underneath the entire recorded history of the country, underneath 400 years of European settlement and the modern multicultural state, there is something older, something that does not appear in any history book, something that can only be seen through the lens of genetics and archaeology and the patient work of researchers willing to ask questions without predetermined answers. That something is the full depth of human presence in Canada. The full 12,000 or 15,000 or possibly more years of human beings living on this land, building cultures and languages and trade networks on it. The people who carried this land inside their bodies long before anyone thought to name it. The Rh negative question is a small window into that full depth. A single genetic marker, measured in a particular country, showing up at frequencies nobody has fully explained. But through that window, if you look at the right angle, you can see the outline of something enormous. The full complexity of what this continent was before Europeans arrived, with their ships and their conviction that history only begins when someone writes it down. The people alive in Canada today carry all of that deeper history in their blood. They carry it without knowing it, mostly because the history is older than memory and deeper than any record. But the blood knows. It remembers. It passes the record forward from generation to generation, patient and silent, waiting for the tools and the will to read it properly. Canada has more Rh negative blood than almost any country in the Americas. The origin is unknown. That unknown, properly honoured, is not a failure. It is a beginning. The mystery is real. The blood is real. Listen. It is still speaking.